Oh, belum di auto sama siapa? Nah, ini tuh tantangan ini Mandiri kalau internetnya. Kopi sungai. Ini udah penuh banget. Sudah jalan. Ini kopi. Lalu semua. Otak siapa? Habisin satu. BIT. Ya itu multimedia. Khusus. Oh, nanti kalau sudah jadi ini jadi masuk mana? Itulah. Oh, ini ada BIT lagi. Kepala BIT. Supaya ini.
Ladies and gentlemen, there are several announcements to all participants. First, have a stable internet connection our session during our symposium. Please make sure that your computer has working camera.
Biasa disconnect ya. Itu ada net di YouTube-nya jarang kan?
Please rename ID according to your category up into the correct in the parallel session. To all participants that would like to ask questions, please state your name, institution, and also the panelist name that you were to question. Ladies and gentlemen, we will begin the plenary session 1. We will have the first session of presentation with the topic of the thalidomide tragedy, importance of applied chemistry, advance in medicine development, Dr. Jahangir Kamaldin. And the second session of presentation with the topic of development of SARS-CoV-2, detection based on RT-LAMP to respond the need of testing capacity in COVID-19 mitigation, Dr. Chandrawati. For this plenary session 1, may I kindly invite our moderator, Dr. Abdi Wira Septama, researcher from Research Center Chemistry. But before Dr. Abdi starts the session, let me introduce our moderator first.
Kalau nggak edit, nggak pertanyaannya. Itu sangat berarti bagi peserta lain. Ya maksimal 12 menit.
Berapa mas? Onisa itu mas?
Ya,
Onisa Triwal.
Dimus
Kepanjangan 22.
Before we start the session, let me introduce our moderator first.
Siapa lagi bang? Mau dicari bang? Nggak ada. Marisa nggak ada. 2000. Dia
Bersama nih, pada umumnya. Ada di Indonesia.
Dr. Abdi got his PhD
Terkirim
in pharmaceutical sciences of Songkla University, Thailand. Master Science in Pharmacy, University Kebangsaan Malaysia, and undergraduate from Universitas Sumatera Utara, Indonesia. Currently, Dr. Abdi is one of the researchers for Research Center for Chemistry. Previously, Dr. Abdi is senior lecturer and Faculty of Medicine, and he got his postdoc fellow in Longhorn University. Dr. Abdi, the time is finally. Dr. Abdi, the time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to all. Thank you so much for MC for introducing me. And from now onward, we are going. I'm going to be a moderator for this plenary session. As been informed, today we have two keynote speakers from two different fields. And I do believe both of our keynote speakers are experts on their own research field. So today, the first keynote speaker is Dr. Jahangir Kamaldin, and the second one is Dr. Chandrawati. And for Q&A section, will be slotted around 30 minutes after the second keynote speaker finishes the talk. For each keynote speaker, we'll deliver the talk around 40 minutes. Okay, let we move to our next agenda to hear the lecture from first keynote speaker, Dr. Jahangir Kamaldin. How are you, Dr. Jahangir? Can you hear me clearly?
Alhamdulillah, thank you.
Okay, before we start our lecture, I would like to introduce our keynote speaker. Can you share the CV? Right. Our first keynote speaker is Dr. Jahangir Kamaldin. It's from Advanced Medical Dental Institute, University Science Malaysia. Dr. Jahangir got his PhD in biology from University Science Malaysia. Got his MSc in biology at the same university, as well as the undergraduate from University Science Malaysia. Dr. Jahangir also senior lecturer at University Science Malaysia. At University Science Malaysia, he is OECD GLP registered lead speaker. ISO 17025 registered lead assessor, ISO 9001 certified leader, and ISO 13485 GMP internal auditor, GCP certified, and the last IOSH certified safety health practitioner. Okay, today Dr. Jahangir will talk about the thalidomide tragedy, chemistry advancements of applied chemistry advancement development in medicine development. For the delay, we would like to invite Dr. Jahangir. Dr. Jahangir, time is yours. Dr. Jahangir, time is yours.
Thank you very much, Bapak Abdi. Let me share my screen.
Ya, harus di-mute.
Pak, tapi kalau buat saya di YouTube aja ya Pak, yang rumah ya Pak. Di Zoom-nya rumah nggak Pak?
Bapak Abdi, is my presentation visible now?
Yes.
Thank you. Okay, Bismillahirrahmanirrahim. First foremost, Assalamualaikum warahmatullahi wabarakatuh to the organizing committee for the very good job organizing this event, which is of course during the still challenging moments. Of course, without forgetting the organizing institution, which is the Research Center for Chemistry, which is now under the rebranding of the National Research and Innovation Agency, BRIN. Of course, not to forget we have Dr. Laksana as the head of the BRIN, and also Bapak Agus, Dr. Agus is now the head of the engineering organization, and of course Ibu Ibu Yeni Emelena, director for the RCC, and of course Dr. Muhammad for the chair for the symposium, and of course not to forget Dr. Tini, our connector, our collaborator, connection between the University of Science Malaysia and also RCC. Okay, ladies and gentlemen, distinguished guests. Okay, I wish to make it as a relaxed session, because we have been very stressed being at home and not able to go to work, do things. As Bapa said earlier, the light can see the light at the end of the time today. We can see more people getting vaccinated and things are getting improved, inshallah. Okay, now fairly to say, I'm very honored to be in this symposium. This symposium consists of all the vast experienced scientists in the applied chemistry or in chemistry. I'm a biologist, but I wish to take this opportunity to share how much I appreciate the existence of chemists, person who do work on chemistry. Without them, we cannot progress. So with the special title today, looking at taking the history of the thalidomide tragedy and how it happened, and how the development of applied chemistry, the progress of applied chemistry become very important in solving or even in overcoming the issues that found, and eventually become advancing in the development of medicine. Therefore, I hope this title meets well with the theme of this so-called theme of this. Sorry, why suddenly it's escaped? Hold on, let me go back. Okay, with the theme of this symposium, which is applied chemistry, breakthrough for a better future. Okay, ladies and gentlemen, I'm as a biologist, I accidentally been working with the chemical from my beginning of my career, from pesticide, from active ingredient, herbal ingredient. So my role basically as a biologist, I evaluate chemicals on the angle, on the perspective to see whether they're safe or whether they're effective in terms of quality assurance. So that's where I believe the chemistry becomes very important. It's prerequisite in any bioassay work. We don't have a chemistry, but you don't have the certificate of analysis of the chemical compound. There's no much fruitfulness in doing the bioassay. So here you can see I cannot start my work even without any chemical analysis. So it comes a rule number one. So therefore, let's look at the agenda of today when I'm presenting how we look in terms of the chemistry in pharmaceutical, in medicine. Now, I'm taking this example from a university in Japan. It has its own department of applied chemistry. And it gives it a definition, of course, the definition. So let me get my pointer right here. The definition is all about understanding the basic chemical properties and eventually producing new materials. It's a new solution. So it is a heavy task. It's not only having a knowledge, but you have to translate the knowledge to something functional. So this is a very hard work to be done. It's a continuous process. And if you look in the daily life, it covers not only the field of knowledge of chemistry, but it relates directly with the, for example, physics, earth science, and not to forget the biology and medical science. So today I will try to put it in the picture to see how much it becomes very important in terms of medicine development. So ladies and gentlemen, if you put applied chemistry, it's very wide, but I want to put in these two very important perspectives, whereby one on I put it my abbreviation CC means chemical characterization. So in my lecture, when I say chemical characterization, it's a very, very long process. It covers identification, quantification, physical, but I just put as CC. And it also have another paradigm of perspective. When you look at HIC, you look at the hygiene from impurities, contamination, which also includes identification, quantification, and removal. So basically, in a medicine finished product manufacturing, these two is essential. You cannot run away. So even Bapak Agus were telling that now we're looking at nutraceuticals. These are key things that sometimes we tend to overlook. That's where, for me, RCC in BRIN play a very important role, and equally in any other institution in the world, the chemists, you are the core. Without you, we cannot move forward. It's very important. So I believe this symposium giving the recognition to that. But I want to give an objective evidence. Is it really true? You see, in the manufacturing of a finished product of pharmaceuticals, medicine, you start with the starting materials. You need reagents, you need consumables, you need standard reference, you need active adjuvant for the production itself. You need to have this identification process, the chromatography, the spectrometry. You need the physical chemical properties. You look into the pharmacopoeia. You see, everything is mostly about chemical characterization, chemical identification, matching with the quality, and so many things. So therefore, if you look in this perspective, sometimes people don't see there's a chemist, there's a chemist life there. Even as a pharmacist, we learn about chemistry. It's the essence of that. It's very important. So this is the background story of how it relates to the medicine development. Now, how about thalidomide? What is thalidomide and why we are looking at the applied chemistry perspective? Definitely, it is a chemical substance, but interestingly, this chemical substance was developed in 1965. The charge number, the system to create a chemical abbreviation system, a chemical abstract system, whereby you assign a charge number, like an identification number to each compound, only existed in 1965. Just imagine all the compounds that was found earlier than that. There's no ID. And this thalidomide was synthesized in 1854, which is almost 10 years before the charge number even been introduced. See? So this is a situation whereby the thalidomide is a very classical story, interesting story, which is just happened after the World War II. But the knowledge of chemistry people translate into medicine. Okay, so thalidomide basically is a compound that interestingly is similar to a biochemical product in our human body. It's a glutamic acid. Okay, and since it's part of the biological system of human, it has its own function. So naturally, is actually an excitation agent. It excites the neuro system, the neurotransmitter in the CNS. Okay, so this was synthesized even in 1954. Dedicated synthesis, not because they want to make it as an experiment reagent or an experiment compound, was designed for the purpose of drug medicine by a company in West German, Chemie Gruntel. So this is something goes back applied chemistry. It's specific. Using a compound with dedicated approach developed into an ingredient for the purpose of medicine. Now, looking at the so-called the development of the applied chemistry, if you see that, as earlier mentioned, thalidomide was developed between this in 1954. The development synthesis started in 1950s and around that. Now, then you compare with the advancement of the isomer chromatography. Okay, earlier, I was in my earlier slides. This is an isomer. It's an isomer. It has its own the isometric component, and therefore at that time you can see how it developed. You see, 1970s. Only then the HPLC existed. Before that, there was no HPLC. So just imagine drug being developed without all these technologies. You don't have the chromatography system in place. All these are not here. These are all critical elements or technology needed for chiral chemistry, but it was not there at that time. Okay, so these are the enablers that will allow. Without this work, these are very hard work done by chemists worldwide, by applied chemists worldwide. that enable new technology which allows better understanding of the active ingredient for the medicine use. Now, looking at thalidomide, the character, right, is a compound that is called racemic. I think you know better than me. But this information was not available at that time. You can imagine with all those apparatus or the technology, they know they have the compound which is thalidomide. They know is a derivative of the glutamic acid, but they might even know the character, might never known the character of the racemic properties of this compound. Looking at, they don't even have the technology at that time. Okay, now why it's become a tragedy? Why thalidomide is tragedy? It's a compound. Now, this the development happened around 1950s, whereby they developed mimics. This compound mimics. Remember, this thalidomide is synthetic, but it mimics glutamic acid derivatives. Okay, and it is specially designed as a sedative drug. After post World War II, people have a lot of stress, people have a lot of anxiety, depression, so they need drugs to make them sleep. So this sedative drug become very important, essential, a good marketing. At that time, it's a very big demand of business. So therefore, there was a push. Why sedative drug had a push? The question, why so many drugs? Why suddenly you look at sedative drugs? That's one of the reasons, because of the post-war scenario, there was a big business opportunity. Again, you can't run away. The basic life of system, there's always a push for demand and also supply. So looking at the demand, so it definitely pushed to the development of a drug for sedative drug. So it was originally meant to give sedation, to enable people to sleep, okay, from the to recover from the depression and so forth. Okay, now in 1950s, it is not common. In 1950s, it's not common for any medicine ingredient, active ingredient. It's not common to test for fetus safety. Nobody do that actually. People don't think about that. So therefore, the thalidomide was never tested on pregnant animals. It was tested in animals for safety, but only on healthy animals. And this is another issue that I want to mirror the current development of nutraceuticals. We rarely see people do investigation. What's the impact of nutraceutical, the natural products on the pregnant model? We never do that. We only do mostly we do on the healthy rats, healthy rabbit, and we declare the product safe. But see, looks what happened. This is the things happened in 1950s, many years back. Of course, that time to blame them, partially is not, because there's no technology available and it's not common practice to do testing on pregnant or on fetus of animals. Okay? So it didn't happen at that time, but they did animal tests on healthy adult, rats, mice, they did that and showed this compound there's no LD50, it doesn't kill. You cannot get LD50 more because this compound is part of the body. It can be metabolized, but again, it has functional, but it's okay for adult non-pregnant animal, no issue. So therefore, it was approved in 1956, okay, to be marketed in Germany at that time. And because the product was so good that it gave a relaxation to people's sleep, it was intended for adults, but nobody considered thinking about issue of what happens if pregnant ladies took this medicine. Nobody really put in the picture that. But not only become a popular sleeping aid, but suddenly in 1961, after only a few years, you see, it was marketed in 1957. Within 5 years, it has to be withdrawn from the market. 5 years. So it went into the market and people found something and eventually has to be withdrawn. Why? What was the issue? Let's look at it. What happened was, it was a good sedative, made people sleep, but not only that, indirectly, when pregnant women took this, it also resolved their morning sickness. Alah mengandung, enggak ada lagi. Morning sickness. Jadi, misalnya, menarik we will take more. So therefore, indirectly, because of these benefits, which is not the original indication, the pregnant ladies took this because it provides them relaxation to sleep. Not only that, relief from the morning sickness, which is for pregnant ladies very important. But at that time when they took, nobody realized, but eventually there was cases happening between October 1957 to 1961. People noticed these babies are healthy physiologically, but anatomically having problem. They don't have they have problem with the upper limbs and the lower limbs being deformed. They're healthy. They can really do their normal life as a children, but they have a problem with this deformed upper limbs and lower limbs. And only within 5 years, they produce about 300 million tablets, and it's affected 10,000 babies. Those survive pregnancy. Okay, so these are the issue, and I took this picture to show now. This is the baby, these are the children, and even until today, they're still alive, but then it's the deformed. They are functional, but only specific is the deformed. So these special changes effect on the fetus definitely goes to the genetic properties during the development. That's where people start to know about teratogenic effect. Okay. So, but my question is that with current situation, we have a very advanced development technology, but we rarely think of this question when developing drugs in current days, especially on nutraceuticals. Okay, we have to go. If you realize the old generation, traditional generation, they have this taboo. You don't take this, don't take that during pregnancy. But we might never know. This was developed based on the observation that it might reflect to the teratogenic effect, perhaps. Now, looking at this so-called, why is that wrong? Because it is part of the body chemistry, biochemistry. Why it become a problem for pregnant women? So just imagine, nobody knew that. Only until 50 years later. Why until 50 years later? Because of the advancement needed on the applied chemistry. A lot of things needed. Without that, we cannot even go. Okay? So therefore, only around 1950s, Handel et al. And they start to discover where it went wrong. So just briefly to share with you, so it's known that actually the thalidomide interacts with the cerebellum, which is a protein component, which is as part of the ubiquitous gas complex that exists in the human gene. It interacts with the human gene, and this expression happens in the brain. Just imagine, at that time we don't have this technology to go to the human genome. Just remember, Human Genome Project happened just a few decades ago, not during that time. Okay? So you need that technology. It means that to understand this, you need the genetic technology to be there. You need the proteomic technology to be there. Okay. So therefore, ladies and gentlemen, and they found out actually, whenever you have this thalidomide interacting with this protein, it also depends with the other biochemicals. What is available? If you have a thalidomide interacting with the protein, but you have another factor, IKZF1 or IKZF3, then you can use this reaction towards the multiple myeloma. This is a cancer. It's a bone cancer. Okay. Now, if you look, if the interaction meets up with the factor of CK1 alpha, then it will relate to this deletion sequence of MDS. This is related to the skin cancer, for example. And if you look into this, now teratogenic effect. So it means that if you need this thalidomide to interact with the cerebellum, with the presence of SAL4, then it will lead to teratogenic effect. Whereas actually, if you have other factor available, you have other two effects, which currently found to be beneficial. This is toxicity, but this is beneficial. See? So this is how it only happens with you guys there. Ladies and gentlemen, you are there, you are here today. You are partial of this changing development for the benefit of the medicine development know-how, but you might never know how it leads. So what really happened to this thalidomide? It's not only thalidomide. There is 1950 story, but actually, if you look at this slide, you see it also happened in year 2000, also happened in 1997. It's ongoing issue. Okay? So it means that sometimes when you develop a drug, due to the limitation of the applied chemistry technology, some of the issue cannot be elucidated. You need the technology advancement going on. So until now, we go to the nanochemistry. Now we go into picochemistry. The more advancement move on, cannot be stopped. We need that in order to further elucidate any potential toxicity, to prevent or to answer what happened before, or even to produce new benefits, to understand new benefits. Okay, so is there any report or any way to prevent this? So because it happened in the 1950s, people had become very this is a tragedy. People become very aware and scared that make things again. So that's where they come with the idea of to manage the development of the medicine based on the pre-market approval. Means any medicine you must have pre-market approval under this quality system. You have the GXP, you have the preclinical development under GLP, you have the manufacturing under GMP, you have a clinical development under the GCP. So it's structured now. Those days you have, but not structured, but now people know a lot of things happening. This is structured now. We have the quality system, but quality system does not have the techniques. You still need the techniques, you need the instruments. So this GXP does not answer the instruments. So we have the system now in place to manage, but we need the instrument to be there. So who developed the instrument? Who develops the applied chemistry? Okay? So I give an example. If you look into the pharmaceutical, we have very different categories. So nowadays, when we talk about pharmaceutical, it's not only the synthetic drugs, even cover the natural products, supplements, even the, of course, biologics. These are not chemical drug alone. It's a hybrid. So things have changed very fast compared to the 1950s. 1950s, when we talk about medicine, we talk about synthetic chemical drug. Things have changed. Now we are in the era of 2020s. You can see now we have a very diverse. This where the applied chemistry development advancement become more important, more challenging to answer this complex nature of pharmaceutical. It's no more going to the small molecules. We have big molecules coming in. So these are very complex. We need a lot of advancement in further to answer the whether to know whether they're effective or they're safe. Now, I give an example. This is like GMP, GLP, GCP, which I mentioned just now, the GCP, GXP. But realize that every this step, you need the chemical characterization, cannot stop. You need, for example, when you develop vaccine prototype, you need to confirm the identity, the biochemistry of it before you can go to the preclinical. When it comes to preclinical, you still need the biochemistry of the what do you call it, analysis of urine, analysis of the blood chemistry. You need to check whether there's a toxic material there. You need chemical characterization. Then you go to clinical trials, same thing. You need. So all the way until the product safety, until the product be marketed, you cannot run away from the applied chemistry being used technology in terms of chemical characterization. It's a very key tool. And this is where all of you play a very big important role towards doing this. Without this, we cannot affirm what we are dealing with. We need to know the identity when we deal with something. So there is a crucial in any process of pharmaceutical or medicine development. So even if you look at people question about the COVID-19 vaccine development, which is fast track. This is the fast track mechanism, which this is the normal mechanism. But actually, they did not move any, still available. All the process still exists. The only difference, they cut short, they allow the overlapping. Previously, you need to resolve one by one, you go step by step. The whole thing takes about 5 to 7 years. But due to urgency, still all they needed chemical characterization. However, they allow the overlapping to happen concurrently to fast track, to answer the issue of pandemic. So, ladies and gentlemen, even though under emergency, pandemic, chemical characterization, we cannot run away. We need it because without that, we might never know what we are dealing with. So the identity becomes very important. But in order to characterize, it's not straightforward. You need the technology. As I say, you need the HPLC, then you have GC, you have the mass spectral coming in. You need all those things in order to. So now we're going more and more high advanced. We're going to nano, pico, need more and more advanced instrumentation relates to the applied chemistry. This is where things happen ongoing, become very important. So ladies and gentlemen, you are the key players in this survival of human to give this example of COVID-19. Now, looking at this, the COVID-19, for example, the development, you see, in the same way, you need to have the quality, safety, advocacy. There's no shortcuts. And as I told you, quality, safety, efficacy, all these stages require chemical characterization using various technologies, instrumentation, which call on applied chemistry. Okay? And again, if you look into the documentation for registration of product, once you complete, this is on the GMP, you complete GMP, you keep the documentation, you complete the preclinical under GLP, you keep the documentation, you complete the clinical under all this you put together, you have to submit for the registration. And all these require documentation on the record, COA, on the chemical characterization and also the hygiene impact assessment on the evaluation to actually is safe for impurities and also safe from any contamination. Okay, now so looking at this is the documentation provided by the international body EMEA. I can just take for example. Okay, these are the various documentation of test method. Each test method, if open up, they will definitely state regarding the chemical characterization, the hygiene, the impurities, and the contaminant issue. All the way, each one, any method you see. So this way, and more importantly, for the purpose of today speech on the lecture on thalidomide, now we have specific investigation of chiral active substance for human. So this is the answer to the thalidomide after more than 50 years. So this is why we have to be cautious and we need the advancement of technology to move forward. So as I say, I'm a biologist, but I can show the evidence and develop. We need to know the safety, but to know safety, we need to know what we're dealing with, what substance we're dealing with. So that's where chemistry becomes important, but not only the compound, but the process of knowing that we need the applied chemistry advancement in terms of instrumentation, technology, and software in order to enable the characterization. So this is the answer after 50 years. Just imagine that. So we might never know what we're doing now may come an issue later, but we still in future technology in order to answer whatever happens nowadays. And again, you see the example for the quality, safety, efficacy in the medicine, they need the pharmacology results, they need pharmacokinetic toxicology. And all this, you cannot run away from from confirming the test item or the active ingredient characterization, the CC, and also the HIC. So you cannot run away with that. Okay, and of course, this is not only issue of one country. We also have this harmonization in ASEAN. We also apply the same. So all the ASEAN countries have accepted this concept, whereby all this documentation of the chemical characterization is mandatory for any registration of new medicine, whether it's a vaccine, whether it's a new chemical drug, or is it national products. Okay, and just to give very quickly, even going details on the pharmacology, these are the data, and then you need to submit again. You cannot run away with the chemical characterization. These are data on pharmacokinetics. These are the data on toxicity, toxicology, and each toxicology needs toxicokinetic. Toxicokinetic, you need the identification, quantification of the compound. Okay? So, ladies and gentlemen, see, but all these people don't. Day to day, general public doesn't know. They do not know how important chemists play the role for the survival of a human in their living, especially in the drug development. And of course, knowing what happened in 1980s, they don't trust scientists anymore, knowing that there are falsification. In US, they found that a huge issue of falsification happening and causing the labs. And this is just one newspaper cutting. whereby almost 4,000 studies were falsified by scientists. So therefore, this is another challenge we have in the development of applied chemistry, is the issue of quality assurance. So that's why we cannot run away from that angle. So therefore, in any applied chemistry works related on the drug development, usually they will require for the implementation of the GLP because of the issue to safeguard any possible possible activities of those that misuse this expertise just to gain the profit. Okay, and even the US law put the requirement on the GLP. So means even though you do chemical testing, you do characterization, when you want to ensure the safety, you must ensure it's also produced. Means the work of the characterization done under the GLP quality system. Okay, and of course another example from UK also. So I just want to give examples, but to show how it relates, to show how important is the applied chemistry until they have very strong law to regulate, to ensure correct true data are provided for the purpose of the registration of the pharmaceutical or in this case medicine. Okay, so even in Malaysia we have the same law. So if you have any drug, any chemical compound that you want to declare as a new active pharmaceutical ingredient or adjuvant or even new biologics vaccine, you still require this requirement of chemical data in order to prove that it is identical to what you're going to deliver to the people when they take up the product or the medicine. And so the question is now, would the applied chemistry as investment that enable the thalidomide to be reassessed or to authorize for treatments? How we want to apply this? Okay, so quickly just to recap again, you look into it happened in 1950s. Those days we only look at the behavioral experiment. Those days, that's what happened when you look at the rats didn't die and there's nothing happened to it. So that's why they're very confident to give to the people to take as a sedative, and even giving to pregnant women, they have no worries. They never seen on the behaviorals. But nowadays things have changed. In 2020s, we are looking at genomic proteomic experiment. So therefore, see, behavioral going to here is not easy route. You need all these examples. Just take a few examples. This is not possible without cell culture media development in 1959. It's not possible without the discovery of this ubiquitin. This discovered in 1965 and further clarified in 1975. This STS page, this is a basic tool. This is biochemical tool. You need this. So this happened in 1970. Okay, and you need the RNA expression. It happened in 1977 and further enhanced in 1987. And more important, today COVID-19 issue. We will never are able to settle this if we don't have an RT-PCR, which we started in 1984. Eventually, we have this. So that's why all this is applied chemistry, ladies and gentlemen. The applied chemistry. As a biologist, I can never do this. But with the help of chemists, we move forward in order to all this development, which now enable. Now the thalidomide can be confirmed the specific pathway, how it can use a hypnotic effect. But the answer now, this genomic proteomic experiment in 1950s is also hypnotic properties, but only based on the behavioral experiment. So you see, ladies and gentlemen, things have changed so big and so much development happened, but bits and pieces we can put together with a lot of importance to have answers to the development of medicines. And there's a specific paper on published showing the thalidomide tragedy out in the 50 years, and there you can see these are research articles. See, this research article, you can see there's a trend going up. See, why suddenly they went up now? And of course, initially, when it's a hot topic, people do research, but suddenly it went down. This is why, because there was not available technology to further understand or elucidate what really happened to that tragedy, the mechanism. But once you have the technology going up, as I showed, from the 1980s, you see when there are more genomic proteomic technology, now it enables more and more research to elucidate. And we have all this now. We have not, as I said, the cell culture, the electrophoresis, the RT-PCR. I think they are the essential tools. But in order to do this, gel electrophoresis, you need chemistry there. There's a chemical reaction there to differentiate the DNAs. So these are things I just want to share with you, ladies and gentlemen, of this auspicious international symposium, which I believe, as a biologist, I myself feel very proud to be here and to show how actually from only one perspective, on the case of thalidomide, how it was very impactful with the advancement. Of course, it took more than 50 years. Perhaps now we can move much faster to answer more bigger issue. For example, like the COVID-19, and maybe the future pandemic that may all come, but with the help of applied chemistry technology, like we know the COVID-19 was very much helpful by the RT-PCR. Without that, we can never come with the vaccine. Okay, and of course, what happened was from that technology advancement, nowadays, even though thalidomide, we knew that it was very harmful, but now with the technology, they can better understanding, they can also use as for leprosy. So it was approved for leprosy in 1998. Because leprosy is a very difficult disease, pathologic disease that needs to be cured, but it seems that the use of thalidomide seems to be very helpful in inhibiting the inflammatory activity. See, that's how I'm trying to put here. If you know in detail, you know how even poison can be a medicine if you know really how to work with it. Okay? And even example now, the thalidomide also be used, been approved in 2006 for the purpose of the bone cancer, multiple myeloma, and also the MDS. So these are evidence-based, based on the capability of the genomic, proteomic works. It's not which requires a lot of technologies, not one single technology in applied chemistry. Okay, and of course, now even there are people talking about whether possible or not thalidomide to use for COVID-19. That's why I picked up this topic, because from something tragedy moving to some benefits, you need the knowledge. So I hope it gives a very good paradigm. See how important the applied chemistry. Something forbidden in 1960, eventually we get to know the actual knowledge, the behind knowledge, with all the applied chemistry technologies to understand. Now we can even to dissect out, yes, this is harmful to pregnant women, but we can use it for this type of disease with a specific care, with the specific design, knowing after we know the so-called the mechanism, which this require high-end technologies of applied chemistry. And therefore, what in conclusion, ladies and gentlemen, if you see the COVID-19 development, we had a lot of issues. Beginning, we started the work, real active work in early 2020. Now we are in September 2021. This is history, ladies and gentlemen. Human have survived. And this survival very much depends on the applied chemistry. Without all those RT-PCR, without STS, without the gene transcription technology, all those biochemical work, without the chemistry knowledge, we may not come today, even though we have so many people died, but we still survive with the help of the knowledge in applied chemistry. So this one to show that how important you are, all of you, for the world of survival of human nature, whereby the applied chemistry is its own field, but the field doesn't stay alone. It's networked to our daily life. And today I share with you only from the perspective of medicine. There are many other perspectives will be shared by other keynote speakers, other speakers. I hope it gives the story how important is actually the applied chemistry in our daily life. So I hope it is basically is that even a hazardous cytotoxic drug nowadays is a cytotoxic. It's a cytotoxic known to be toxic, but can be safely manufactured, distributed, administered to patients during chemotherapy. And now we even know how to dispose responsibly. So who does this? Basically through the applied chemistry for the for a better future. I hope with this sharing, it gives some an idea, a small sharing that telling how important is this field. I hope you all will progress, success. We need you for the survival of human nature. With that, I'll end my session, and my apology if some of my deliveries was not accurate and so forth. I hope it achieved the main intention. And again, thank you very much for the organizer and for this wonderful session, bringing together all the expertise in applied chemistry and chemistry, perhaps also from other field, pharmaceutical, pharmacology, and also biochemistry. So with this, I pass over the session to Bapak Abdi. Silakan, Pak.
Thank you so much, Dr. Jahangir, for your nice presentation, your valuable information. What I can say that from tragedy we can find a new insight, and by combining not only one discipline knowledge, but we have to combine all multidiscipline knowledge, just like biology, chemistry, pharmacy, and many knowledge. I think all the participants must have the question. So you just drop your question on the chat room, and we will discuss after the second keynote speaker presentation. So let me move to our second keynote speaker. For the next speaker, we have Dr. Chandrawati. Could you please to share the CV? Well, before we proceed to our second lecture, I would like to introduce our second keynote speaker. Is Dr. Chandrawati, our senior researcher and research center for chemistry. Dr. Chandra got doctoral degree from School of Pharmacy, ITB, in field of pharmacology, master degree in biochemistry from the University of Nancy, France, and undergraduate in biochemistry as well from the University of Nancy, France, and general university degree in science of nature and life from the University of Nancy, France. Currently, Dr. Chandra also have research grants. She is a principal investigator for the project about detection of RNA virus of SARS-CoV-2 based on heart turbidity, and grant from LPDP, Ministry of Economic, and also got a grant as a principal investigator for the research about development of natural compound silvestrol as drug candidate for hepatitis virus and emerging virus, RNA virus. A research collaboration between LIPI, Rural University Bochum, Department of Molecular Medical Virology, Institute of Organic Chemistry, Leibniz Hanover, and grant from BMB Germany. And last, formally he got grant from German Indonesia Anti-Infective Corporation, a systematic approach for discovery of novel anti-infective with emphasis on viral metabolite from terrestrial Actinobacteria, Myxobacteria, marine bacteria, and their infected hosts. Without further delay, I would like to invite to Dr. Chandra to give the Dr. Chandra, time is yours.
Thank you very much for the nice introduction. Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to thank the organizing committee for inviting me to this conference. It allows me to meet all of you to exchange our knowledge and experience. It's also a great pleasure to me to have occasion to share our experiments in developing SARS-CoV-2 detection based on RT-LAMP. I hope that the topic is in line with the theme of the conference this year, Applied Chemistry Breakthrough for a Better Future. That we also hope that what we are doing may contribute to bring solution to our current health problem. So may I share my presentation? Okay. So the talk that I have today is development of SARS-CoV-2 detection based on RT-LAM to respond the need testing capacity in COVID-19 mitigation. As we know that SARS-CoV-2 or severe acute respiratory syndrome coronavirus 2 is a virus infection that causes respiratory disease, coronavirus disease 2019, which can be fatal and also can lead to death. So due its rapid and widespread throughout the world, in March 2020, WHO declared the status of COVID-19 as pandemic. Until now, efforts to find effective vaccines and therapies are still being carried out around the world. And also in Indonesia, we have strategic efforts that we have already started to control the spread of the SARS-CoV-2 virus, and also to reduce the death rate from COVID-19. According to the WHO, the strategy that needs to be considered to stop the spread of virus is to develop the ability to conduct rapid and accurate screening and diagnosis to find out whether a person is infected with SARS-CoV-2 virus, isolate that person, the positive person, and conduct the history tracing of anyone who has contact with the person to be subsequently quarantined for surveillance. So this is called 3T, namely testing, tracing, and treatment. This is that the effort that Indonesian government already adopted since the beginning of the pandemic. Currently, the ability to perform testing in Indonesia is still low. Yeah. Okay. I'm sorry. I forgot to give the outline of my presentation. First, I will show you what is the background of our works, and then the basic principle of the RT-LAM method, the design of RT-LAM method, the RT-LAM, and the characterization of RT-LAM, and the end is the conclusion. So the first background, those are factors that motivated us to conduct the research to develop the detection system of SARS-CoV-2 virus. Of course, because of the pandemic, that until now we are still undergo the research to develop the efficient drug and vaccines. And also, we also already adopted the mitigation strategy, testing, tracing, and treatment. Currently, in the ability to perform testing in Indonesia already reached an average of 24 to 34,000 people per day, but this is still far from other countries, especially the developed countries. So, but not only the problem of the quantity, but also the most important is the covering size. I mean that the testing is still not cover a large area, because we know that Indonesia is a big country. So the testing should reach all corners of the countries, and also the problem of accuracy, because this needs a technology that is not more advanced technology. For testing of SARS-CoV-2, currently we have a golden standard that we call qRT-PCR, or reverse transcription polymerase chain reaction. That is a molecular-based test that is able to detect the presence of the virus sensitively, specifically, and accurately. This is using the samples from throat, nose, or swab. And then, but however, this technique has the disadvantage that is relatively expensive. And then also this technique has limited application because it requires lab facilities and then the chemical equipment that is expensive. The regions itself also expensive. Here also we have a problem that all regions actually are imported. So this causes the problem of bottleneck when the need is high because it's we depend on the producer regulation, yeah. And also the urgency of the test is high, especially when we are in the peak pandemic. But even when the pandemic is decreasing, the need of testing is still important because we still have to control and to check the spread of the virus itself. And then again, the need of applied technology. This, as I mentioned before, that to be able to have a technique that is accurate, we need an advanced technology that we need to acquire new technology to improve ourselves, yeah, for the future, that we are more ready to face another outbreak. So for those reasons, we need to improve our testing capacity, and we have an idea to develop detection, RNA detection based on RT-LAM method. So the background, okay, LAM or loop mediated isothermal amplification first is introduced by here Notomi from ICON that is the method that can synthesize DNA with high specificity, efficiency, and rapidity under isothermal condition. So this is in 2000, already a long time, but the use is still limited because we have already know another technique, RT-PCR, already applied worldwide. So according to this paper, the LAM reaction needs the DNA polymerase with the strong activity for strand displacement. And it requires some set of primers, different as PCR that only need 2 primers. Yeah, this need the LAM method needs 4 or even 6 primers that recognize the sequence on the target DNA. And then because the LAM reaction use more primers and more target recognized, that makes the LAM reaction can amplify the target sequence with high selectivity instead of 2 in the case of PCR. Yeah? And also according to this paper, the formation of loop that makes incubation occurs in a constant temperature. It's between 60 to 65. So we can only use water bath or heat block. So this method can be performed in regular laboratory. Yeah, this different technique that already most of those is different from the detection system. So this, for example, this we can detect the amplification of the sequence targeted by change of fluorescence, and here by the change of the color by colorimetry, and also possible by electrochemical, and here also targeted fluorescence. Or we can also develop with the lateral flow in the assay. Yeah, it's already some groups already developed this and already in market, already in use. We also can use the product of LAM reaction by electrophoresis, and also that this what we developed in our laboratory by detecting the precipitation of magnesium pyrophosphate that we can observe by naked eye or by real-time turbidinometry. Okay, so nowadays actually use the LAM reaction is already used in different cases. For example, in plant virus, also in foot and mouth disease virus, it is in animals. Also already established to detect MERS-CoV COVID, or the chikungunya virus, and also multiple influenza virus, Zika virus, and lately SARS-CoV-2. That the technique is still growing very fast. Now we have a lot of kind of SARS-CoV-2 detection based on LAM with different technique of detection. Okay. And then the basic principle of LAM here. Okay, this look complicated, but actually it's because we use six primers in pair. So in the end of 5 prime, sorry, in the end of 3 prime. Okay, so actually we first of all, the LAM reaction is divided in 3 processes. So the first is the reverse transcription itself, because SARS-CoV-2 is an RNA virus, so we use only a single strain. So it has to be converted to DNA by the reverse transcription. And then the second one is the basic structure formation, that this as template the structure template to do amplification. So this structure will be amplified several times, several cycles, to have amplifying the targeted sequences. So actually we have here, yeah, the couple of primers, but I only call this inner primer, external primer, and loop primer. So BIP is internal primer. So first of all, the internal primer will go to the target sequence to do a reverse transcription and to do the second strand. And then the other side, there is another internal primer with that will synthesize from the other side, and then that can make the loop for the next polymerization. And then here we have the single-stranded cDNA synthesized from the inner primer, that we have now 2 strands of DNA. So we call that cDNA. And then here from the cDNA we continue to the polymerization by the second primer, the external primer F3 and B3. Here the first F3, and then the second one B3 will come to synthesize another N. So this the most important of the LAM reaction is once one strand is synthesized, the former strand is displaced. So there is no need to do the denaturation to discard the two strains, because the polymerase has strong activity for strand displacement. So here, after the external primer synthesize the cDNA, we have the structure like that. We call that the dumbbell-like structure. this is actually the seed structure for the next step, that is amplification, that will be amplified several times. So here there are some points of initiation of polymerization, yeah. Not only because to do polymerization, the enzyme need a single strain. So there is internal, external, and then here the loop comes. So the loop is here is the single strand part, yeah. So that also possible to initiate the polymerization. So here comes the loop primer, and this also makes the amplification more improved. Yeah. So after that is a cycle. So this the three primers come one after another, and at the end of the reaction we have fragments of different size. We call that amplicon, that the fragments amplified during LAM reaction, that we can visualize under electrophoresis or other machine, yeah, like turbidimetry or colorimetry or fluorescence. So I put again the figure here because I would like to put side by side with the RT-PCR technique. The big difference is that in the RT-LAM, the reaction occurs in continuous. There is no need to hit the reaction to discard to separate the double strand. But here in RT-PCR we need some temperature cycles. For example, here for the denaturation we need to increase the temperature to 96 in order to separate the double strand, and then decrease the temperature to 50 to do annealing of primers. There are only 2 primers here instead of 6 primers in RT-LAM. And then here, after that, after annealing, then the enzyme polymerase comes to do polymerization. And this occurs at the temperature 72 degrees. And then after complete polymerization, then the reaction needs to be, the temperature needs to be increased again to 96 to separate the strands, double strand, and this will happen in cycles. And normally we need until 40 cycles of different temperatures. Yeah, so why RT-PCR? We need a machine that is sophisticated, and it costs very high. It's expensive. So more than 500 million, I think. Yeah. So this different. So here RT-LAM only occurs in constant temperature for polymerization, and for RT-PCR we need the cycling different steps of different temperatures. So we at the beginning in our group we choose to develop RT-LAM with turbidity detection, because we think that this system needs to be used in regions that are very limited in facilities, laboratories facilities, and we hope that we can develop a system as simple as possible to be able to be used for a lot of people. So turbidity is our choice. The best method of turbidity is because during polymerization of the targeted sequence, the enzyme,
Di bawahnya.
add the DNTP to the targeted, and here, by inserting one nucleotide, and it release this pyrophosphate ions. And in the solution, we add magnesium sulfate. So the reaction between magnesium sulfate and pyrophosphate give the precipitate magnesium pyrophosphate. That is the white precipitate that we can see here very clearly after the end of reaction. So this, for example, when we have samples, then we only need a water bath that is set for 60 to 65 degrees Celsius constant, and to allow reverse transcription and amplification of the products. And here, as the sequence targeted is amplified, the magnesium pyrophosphate is accumulated and give the peak, the peak here after sometimes, usually after 30 to 50 minutes. Here is 20 minutes, depending on the quantity of the DNA used, and also the CT values of the virus load. And here is the picture of the gel electrophoresis that we can visualize the product of the amplification of LAMP. Here we can see that the bands, the fragments here, we see as the bands that seems like leather. So this means that the fragments are amplified comparing to the control. Here is clear, no fragments amplified. So this also can be proof that the reaction LAMP is positively, the target is detected. Okay. So what is our Artilan design in LIPI? We have already designed our system. First of all, we have to choose what gene are our targets. Here, as we can see the structure of the virus, here we see that there is a protein here, a spike, the membrane protein, and then we have envelope, and then we have capsid protein and envelope protein. So here are the genome of the virus. They have around 30,000 kilobase. So this is the genome of the SARS-CoV-2. This is SARS-CoV-2. We compare to the MERS and the newly this is SARS-CoV and this is SARS-CoV-2. Yes, and we have to choose the region when there is no variation. Yes. Here we choose the origin. The target is the ORF, and also here in the N protein. So those are gene targets that are already used. This according to WHO, so already submitted to CDC. And for example, China, they use ORF and N. And then France, 2 targets in RDRP. There's only 1 gene, but 2 targets in the gene. CDC use only 1 gene, but 3 targets in the N gene. And then Japan, spike protein, and then there are also countries using three different genes. and also countries that only use one gene. So here we, in our group, we choose this gene, RF1A, and then the gene N, that we look for the sequence based on the genome sequence already deposited in NCBI. So from this gene, we choose, we start to design our primer, which sequence from this gene are best for the primer construction, as well as for the gene N. Oh, sorry. Okay. Oh, sorry. So we are here. Okay. Okay, I'm sorry. So then we start to design our primers. We have to consider some points. For example, when we need to design primers, we have to look for the distance between primers. That, for example, for the internal primer, the distance, here. Yeah, we need to make sure that the F2 region and the same sequence as the F1C region at the 5N. There is a distance, a specific distance that it's allowed here. For example, the distance is between 120 to 180. The F3 primer is the external. I call this external primer. This is internal primer, and this is more outer region than internal primer. Also in the other side, and then F3 is coupled with B3, and then this is external, and then FIP, internal primer, is coupled with BIP, see the other end. And in our group, we also add loop primers. This is actually additional, but we think that by adding loop primers that we add one more region targeted that contains single-stranded, that could increase the number of starting points for amplification. So here we also design the loop primer here and here. So this is the main points of primer design, as I already mentioned before, that for example, the distance between F2, this is F2 and B2 is internal primers is between 120 and 180, and then this is the external primer is not more than 20 base pair, and the loop forming region is between 40 and 60 base pair. We have also considered the melting point value for primer regions, that in case of GC rich, the TM should be between 60 to 65, and for the sequence with 80 rich, the melting temperature should be between 55 to 60. We also have to consider the stability of primer. We have to calculate the degree of free energy, DG. This not more than 4 kilocalorie per mole. So for each couple, and also we have to consider the GC contents. We need the GC content not more than between 50 to 60 for the GC rich and normal, and GC content is about 40 to 50 for the 80 rich. And we have also considered the secondary structure, because this has to be stable, because otherwise the primer will be very easily broken, denaturated. So Oh yeah, I'm sorry. There is a software to design primers. This primer explorer. So sometimes we have to try and errors and then check and then also check in the laboratory whether it's we have to also to optimize the reaction. So what we have already done in the beginning. So we use two enzymes, the polymerase with high strand displacement activity, that is BST polymerase. This is from Bacillus thermophilus, and this enzyme contains the 5' to 3' polymerase activity, but lacks 5' to 3' exonuclease activity. So we use this, and then we use reverse transcriptase, this from avian myeloblastosis virus. And this to synthesize cDNA from the RNA or single-strand DNA template. And then this is our Actilam design. So once we have already designed our primers, and then again changing target, and this the LAM reaction. First, in the beginning we use both, water bath, also heat block. We also check with fluorescence because we also have the machine. So not only we detect the precipitation, but also the fluorescence. We add some fluorochrom on the reaction, and we check with our thermocycler. And this is what we observe after the Actilam reaction, that we can see the formation of the precipitate. Here is trouble, or is we see the precipitation in the positive sample, and no precipitation in the negative sample. Here, when we centrifuge the solution, we can also see the pellet, and in the bottom of the pile, then you can observe that there is a pellet in the positive samples and nothing in negative. After centrifugation, we also can use ELISA rider or spectrophotometry to detect the turbidity after following LAM reaction. So here comes a characterization of our Actilam design. So this is what we have done, that we need to have the swab sample, and we have a collaboration with the what we call it, health agency in province, Banten province, that they supply us the extracted RNA, and then we only do optimization of the LAM reaction, that we only use the incubation in the same temperature. And also we also in collaboration with our colleagues in the physics department, that they develop real-time turbidimetry that allow us to control or to follow the LAM reaction with this machine. So this is what the reaction looks, that after sometimes in 25 minutes, that we see that there is an increase of the amplitude. That means that there is formation of turbidity, that the salt, magnesium pyrophosphate. This, after centrifugation, we also check that there is a pellet and nothing when the sample is negative. So this can be detected real time. Okay, so first of all, before using the machine from our colleagues from the physics department, we check the machine comparing to the our thermocycles, our fluorescence machine. So this gives the results approximately the same at the time around 30 minutes. You can see the amplification of the samples, the positive samples, and nothing with the negative samples. It's in line with what we observe in fluorescence. So this means that we can use the machine, the real-time turbidimetry for our experiments. So, and then the second phase is we have to validate our system, because we have designed our primers, and then we choose in order to optimize the reaction, we have to validate. We use the synthetic DNA. This synthetic DNA means that we synthesize the sequence that I showed you before. The gene, the N gene and the RF gene, we use the plasmid PUC57 here, and then we insert our genes RF and N here, so that the total length of the plasmid is around 3,216, and by electrophoresing, we can see that we have the plasmid containing our gene that we use as the sample for our RT-LAM, for our LAM reaction. So this is what we got here. That with real-time turbidimetry, we see that the positive samples, there is an increase. That means there is an amplification thing with the template recorder. And as well, we observe there is this becomes turbid when the sample is positive, and nothing when the sample is negative. Also, we observe the pellet, and the electrophoresis picture of our product, of our LAM product, that there is, you can see there's a ladder for the positive samples and nothing for the negative samples. That means that we have already validated our system, our RT-LAM system, that we can move to do some tests with the sample from the health department from the Banten province. We use arana extract from swab nasofaring, and we also compare to the golden standard PCR with target. They use helicase. So these are some results. We use different city values that we obtain from them. Here with the city values 21, we still see we can see amplification with our RT-LAM system. Also with city 25, 30, 32, 35, 34, and here with city 36, there is no difference. So we cannot observe the amplification by our system RT-LAM. So that means that our limit detection is city 36 compared to the golden standard RT-PCR with target gene helicase. And also we look for the quantity of total RNA extracted, that we vary concentration to see the limit of detection. Here below 40, 40 nanogram, we can still observe the pellet. That means it's still positive. So, but when the quantity is 12.5, is cannot see the amplification. So the limit, that means that our limit is around 40, above 14 nanogram. So afterwards, we look for the specificity for our ORF gene that we have already chosen. Using in silico, we compare with the database sequence. We use database from NCBI and GSI. And here we can see that, this is the representative of the different regions in Indonesia, also in Asia, in Japan, in Australia, Europe, America. So it's only representative that we found that almost 100% of homology gene target. So that means that our system can recognize the different variant of virus from these different countries. So that means that there's no problem where the virus comes from Indonesia or other countries, that our system still recognize the target. And this is the cross reactivity. We use the sequence from the pathogen microorganism that we have in our respiratory tract.
Excuse me, Dr. Chandra. You still have 5 more minutes.
Okay, I think this is nearly the end. So that means here with this pathogenic microorganism is not compatible. So our alarm system cannot recognize this sequence. That means it's specific to SARS-CoV-2. So this is also for gene N. This is the specificity test that we conduct with the comparison with the artificial that we do in our lab with the same target gene of IF that is totally the same, that we also found the same results. So this is that means that the sensitivity is 1, specificity is 1, and accuracy is 1. So this is the conclusion. This is actually our system with the reaction condition, the sensitivity, comparing to the CT of RT-PCR, and then comparing to the amount of RNA, specificity, and cross reaction. That means that our RT-LAMP system can be used as an alternative method of RT-PCR for detection of SARS-CoV-2, which provides a rapid, sensitive, specific, and simpler and less expensive procedure. Thank you so much for your attention. If you have any question, I will be very happy to discuss with you. Thank you. I'll pass over the mic to the moderator. Thank you for the time.
Thank you so much, Dr. Chandra, for the nice presentation. I think now we have the alternative tools, that we can use to detect SARS-CoV-2. So we don't have to depend on the RT-PCR or something. We can use any tools that already developed by our researcher in LIPI, in Dr. Chandra. Okay, let me move to the Q&A session now. So here we have one question from Dr. Yeni Meliana. To Mr. Jahangir and Bu Chandra, what is the biggest challenge when you start your research? Maybe Dr. Jahangir can answer first.
Thank you very much, Ibu Melena. Okay, good. I think this question is relevant to any researcher. Of course, for me at this age, after doing many years of research, that challenge will be less. But I think the current challenge. What is the current challenge about this? That previously, when we embark with any research, people will ask question: what is your publication? How many papers you will publish? That was those days when I was a young researcher. Unfortunately, I believe with the new generation, they have a bigger challenge. Now the question is how much money you can make from this. That is, I think, the current shift of paradigm. Those days more about how many papers, what is the impact factor of those days, and no, but now the question comes is that when we even start to propose, people ask, what is the outcome in terms of the money? So I think this is where for this generation, you maybe not necessarily should be young, but in this generation of research, this is a challenge. This is where the know-how of the past research very important. But we cannot spend too much on doing the basic research, but we have to network with the information from others to make the foundation of the basic information so that we can move to the next step. So means that I believe to overcome this challenge is to network with the right partners, and get the basic data and show how we can move forward rather than to say I will do everything. So means we have the basic data, put it together. Like now, I think Dr. Chandrawati have an example how you move forward. You have the background information, background technology, you move forward, and perhaps from there we can show how we can produce the money, even though we are not a salesman. Okay, so I hope answers that. Thank you.
Okay. My turn? Okay. Yeah. Thank you, Bu Meli. I think it's a good question. Yeah, because it's our concern as a researcher. I think it's always our problem when we start any research. Yeah. So for the research of COVID-19 at the beginning, it was very hard because it was the peak pandemic that everybody has working from home. I think that the beginning is only the teamwork. Yeah, first of all, it's teamwork that we found colleagues that have some the same vision, same ideas, and knowledge. So we start to design the research only small people. Yeah, so. But afterwards, the teams are growing, so we can also include the partners, other partners. This is at the beginning. So first of all, teamwork, and then the second, I think this is very important because this can overcome other problems. This management support, because this is very important. I'm very happy that I have the head of our office is very supportive since the beginning. So with her help, that we could get the finance, we could get the lab facilities, and also everything that we need to perform our research, because of there is a management support that becomes there's always solution. So this is what we have experienced from the beginning. Yeah, so teamwork, management, facilities, and finance. Thank you.
So how is the answer, Dr. Yani Meliana?
Yes, thank you. Thank you for the answer, Dr. Jahangir and Bu Chandra. Thank you.
Yeah.
Really great work.
Yeah.
I have to thank you.
I think this example symposium is a platform we create the teamwork. As what Dr. Chandra says, that everybody here, this is where you start to get to know each other and teamwork. So I think this is a very good platform for everyone, not only in Indonesia, in ASEAN, but perhaps the world, to network and to know how we can help each other to develop a much bigger outcome and solutions.
Yeah, I agree. Thank you.
Let me move to the second question from Devi Universitas Negeri Jakarta to Dr. Chandra. What do you think are considered as contaminant in the experiment, since this experiment is quite sensitive, and how to overcome if there was a contamination in these experiments?
Okay, thank you. So this is by our primer design, because we have the primer which are very specific to the target. So as we targeted the SARS-CoV-2 virus, then we design our primers only recognize this type. So we have already checked with the in silico and also comparing to the other methods that there is nearly 100% of good recognition. So not another virus, because we have also checked with the cross reaction with other microorganism in our respiratory tract. So there's no match. They don't match. Our primer don't match to them. They only match to SARS-CoV-2 virus.
Okay, Haris Devi is the answer. Okay, maybe I will ask Dr. Chandra. Still related with this question. In your slide, you stated about among there are four, if I'm not mistaken, 4 systems that can be used to be used in alternative detection, SARS-CoV detection. First, colorimetry, fluorescence, turbidimetry, and the last one, lateral flow, something like that. I forgot. Among this detection system, which one is more, is more powerful? And you can explain a little bit information about this. Why you choose colorimetry and then?
Yeah, thank you for the question. So actually, there are some advantages and disadvantages for each method. Yeah. So we have at the beginning of the experiment, we have to consider some aspects like simplicity and not very expensive, but still accurate and specific. So actually, the other, for example, for fluorescence, I already show in the slide that they are actually more powerful than our turbidity, but still very good, because it's fluorescent, they can detect more than CT36, but for us, CT36 is already not contagious, yeah, so not infectious, so it's still acceptable. And with the machine that the facilities we don't acquire expensive facilities, so I think turbidity is more economic than the others, and we still can do some quantitative detection, because we also develop a machine, but the machine itself is actually very simple, because they don't need filters, no need, only lit LED, that's all. So, and with this machine, we can already follow the lump reaction in real time. So, yeah, I think is why we choose this method. Yeah.
Okay, thank you so much. Any question for participant? You may ask our keynote speaker straight away. No. Oh, I have one question to Dr. Jahangir about adulteration. As we know, that we don't have a universal guideline that can be used to standardize. If we let's say in China, maybe in China they don't care about any adulteration on their natural product or drugs, material or something. But in Europe, they are very strict about the contamination, adulteration, and something like that. Is there any universal guideline, harmonized guideline that can be adopted all all the country all around the world. So how to say, we can accept all, not part by part, something like that.
Oh, thank you very much. I think that's a very relevant question about adulteration. If to test a product for the entire possible adulterants will be almost impossible or not cost effective. There are hundreds of compounds that you can adulterate. So what the regulator has done is to relate with the quality assurance mechanism of GMP, good manufacturing practice. So that's where when you want to produce a product, a medical product, pharmaceutical, it can only be produced in a GMP. I think in Indonesia, in Thailand, Malaysia, even in China, they already started this already, whereby only certified manufacturing. And in this certified manufacturing, they can only purchase the active ingredient or the excipient from certified manufacturers. So therefore, the manufacturer also have to produce under GMP. See, the manufacturer of the active or so-called the ingredient, the excipient, also have to comply to the pharmacopoeia, either US pharmacopoeia or European pharmacopoeia. So this cascading effect, in a way, helps to prevent. May not be 100%, but helps to prevent. So therefore, the control from contaminant and also from because sometimes the adulterant could be from contaminant. You don't realize it. Actually, you don't purposely put that, but somehow it was in the product due to the natural occurrence. This thing happens for our athletes. When they go for the testing, urine test, they fail because they take herbs. But they didn't know the herb also have the steroids, accidentally, not purposely perhaps. So therefore, the way of doing that means resourcing the ingredients specifically from the reputable or certified manufacturers. From there, you reduce the chances of the adulterants by virtue of on purpose or accidental. Then it goes to the manufacturing of the finished product, also by virtue of control. And each time we have the COA, so that COA will tell you the impurities. When you saw the impurities, that gives some idea how much the adulterant will be there. So of course, in a specific manufacturing finished product, they have a target possible adulterants. So this depends on the country. We cannot put universal. For example, in Malaysia, there could be list regulated. They say these are the list of adulterants. In Indonesia, list by means of the priority and also by list of the geographical situation. So therefore, there are total list of adulterants that commonly found that been published by WHO and ICH. There are. However, for economic purpose, we bring in together with the GMP mechanism, together with the chemical analysis to reduce the risk of adulterant. So, but I think, so for my experience, we have seen this happening under the Pharmaceutical International Convention, PIC, already done. So all the countries there. So actually, ladies and gentlemen, maybe not directly from analyzing, we don't do the mass spec for the entire adulterant, but we do the combination between the administrative control by certification and also using the chemical analysis with targeted. I hope it answers. Thank you.
Okay, thank you so much, Dr. Jahangir, for your explanation. Yeah, clear already. Okay, is there any question for participant? We still have 10 minutes for discussion.
Bapak Abdi, can I share something?
Yeah, sure.
Okay, ladies and gentlemen, when we develop new findings, new product, new solution, definitely there will be some form of uncertainty. We try our best to be confident, but we know there will be some portion of uncertainty there. So what happens is that we shall develop what we call it quality assurance or quality control. For example, any detection kit, any method of detection. If you remember, you always have some control to put there. To say calibration curve, all those things. So don't worry. As long we have this bill in mind about the quality control, we will reduce the uncertainty. Things might happen. We might never know because whatever we develop in the laboratory or the field trial is still limited. It may not answer for the entire world. But nevertheless, we will progress. It's not one stop. Once you develop a solution, there will be progressing quality control and slowly. For example, you have started to have use in Africa, for example, to be different domain, different, but we start to develop the quality control. So that's how it works, gentlemen. So for everyone, don't worry that, don't be very be worried that people are raise question that are you sure I certain. These are normal. That things happens by progressively. But what important, we take note there are uncertainty, but we build in mechanism to how to manage the uncertainty. For example, we have controls, positive control, negative control in our mechanism and our process. So definitely in science, we don't say 100%, but we go towards that by having all this mechanism. As I say, it can be from the manufacturing, from the end user, many ways you can do the technology. So that I just to share for everyone, we should progress and be bold enough to come up with a new solution, although we know there still are uncertainties. Thank you.
Okay, thank you so much, Dr. Jahangir. I think time is over already. Thank you so much for our keynote. Oh, sorry, we have one more question from Haryadi to Dr. Chandra. Sorry. In terms of selectivity and or sensitivity, the turbidity may not be compared with fluorescence, but for prick detection, it might be very useful as quick method. Any comment for this, and how will be this approach will be developed for future at the same research area? Okay, you can answer the questions right away. You still unmute, Dr. Chandra.
Okay, I'm sorry. Okay, thank you for the question. So actually, we have to distinguish two aspects. Yeah. So first of all is the LAM reaction itself, which means that the LAM reaction is to target the gene and then to amplify, to be able to detect. And then the second one is to detect the amplification results, to observe the amplification reaction. So if we talk about the reaction itself, it's when it's the system is good, then it's good. So the difference is in the detection. So we can add a fluorescence in the reaction. So because I also do that, and also there is also in the commerce. To check the system, if our system LAM is correct or not, and then with other detection system should be should give the same results, because it's only the matter of detection. So I think what different is when we have the different targets first. Because if we compare also with PCR, it's the same principle. So I mean the method is the same, because we target the certain sequence and then amplify and then observe. And this is the same method, yeah, the same basis. So I don't know where is the problem, because we can also compare. RT-LAM and RT-PCR we can compare, because it's different principle with different basic, yeah. But with other, like for example genos or for example antigen, the target is different. So it's not the based on RNA, but the protein. So this may be the protein or other aspect, yeah, like genos is they detect the volatile compounds, yeah. So this may be different, but fluorescence and RT-LAM they are the same methods. So I don't see I don't see the problem. Thank you.
How is Mr. Haryadi? Is the answer?
Yes. Thank you very much for the clear answer. Thank you.
I think that also we finished already. The time is over. Please big hand to our keynote speaker, Dr. Jahangir and Dr. Chandra, for your informative, your followable sharing with us. Hope to see you again. Stay healthy and stay safe. And for the next, I will give back to to Dr. Zetri, his MC, and for the agenda. Thank you so much, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you to our speakers and moderator for very informative sessions. Ladies and gentlemen, we will move to the next session of award certificate. As an award from the ISEC 2021 committee. This certificate will be given to Dr. Jahangir Kamaldin as keynote speaker of plenary session.
Thank you.
Thank you, Dr. Jahangir.
Dr. Chandrawati as keynote speaker of plenary session 1.
Thank you so much.
Ibu Chandra.
To Dr. Abdi Wira as moderator for plenary session 1.
Thank you so much.
Terima kasih, Abdi.
Ladies and this is the end of plenary session 1. To all participants, now we will have break time from 11:36 until 12:45. In advance, we would like to make an announcement. There are two parallel sessions to be conducted. Parallel 1, Wednesday, September 29, 2021 will be started on 1 until 3 p.m. And parallel session 2, Wednesday, September 29, 2021 will be started from 3:15 until 5 p.m. There are four parallel breakout rooms in this Zoom meeting. Parallel A, green process and chemical engineering, clean energy production technology. Parallel B, material and catalysis. Parallel C, food processing technology. Parallel D, pharmaceutical technology and drug delivery system. Natural product, medicinal chemistry. Parallel session 1, room parallel A will be moderated by Deliana Dahnum PhD. Room parallel B will be moderated by Siti Nurul Aisyah Jeni PhD. Room parallel C will be moderated by Anastasia Devi PhD. Room parallel D will be moderated by Faiza Mariani MSc. Day 1, parallel session 2. Room parallel A will be moderated by Roni Mariana PhD. Room parallel B will be moderated by Saiful Rahman PhD. Room parallel C will be moderated by Hakiki Melanie PhD. Room parallel D will be moderated by Dwi Antika, MSc. We hope all the participants will join in each room until the end of. All participants will automatically enter each breakout room according to the schedule in the program book. Please ensure your Zoom ID correctly written according to your participant category. I hope the participant will have a great time for the rest of the symposium. The plenary session 2 will be held tomorrow, September 30, 2021 at 8 a.m. Ladies and gentlemen, the 7th International Symposium on Applied Chemistry 2021 is sponsored by Era Mitra Perdana, supported by Dynatech International, Horiba Indonesia, and Berka Niaga Mitra. As Master of Ceremony, from the deepest my heart, I do apologize for my mistakes in presenting this ceremony. Thank you very much for your attention, and goodbye. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
I would like to make announcements to all participants. The committee already renamed the Zoom ID according to the participant category. Please ensure the ID is correctly written within the format after you re-log in into the Zoom meeting room. Thank you very much.
Thank you, Zetri. Okay, thank you everyone. Dr. Jahangir, once again, thank you. Stay healthy. We will meet again next time, okay? Face to face.
Thank you, Dr. Jahangir.
Mobilitas tinggi di setiap detik dan visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi inovasi membangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur daya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Indonesia berdaulat, maju, adil, dan makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, 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 mobilitas tinggi, kebutuhan yang meningkat. Antara akademisi, pebisnis, sinergi standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era akademisi, pebisnis, sinergi antara akademisi, visi 2045, Indonesia Berdaulat, maju, adil, dan makmur telah dicanangkan. Sinergi antara akademisi, prestasi, dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sinergi antara akademisi, pebisnis, sinergi antara akademisi, 9 fokus riset telah ditetapkan. Sinergi antara 49 produk inovatif telah direncanakan. Sinergi untuk transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern, mempunyai inovasi di mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, maju, adil, dan makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian sangat penting di sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Dengan pemberian riset dan inovasi adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokuskan koneksi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi kebutuhan melahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat perusahaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Riset dan teknologi, Badan Riset dan Inovasi Nasional adalah tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, melahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia berdaulat, maju, adil, dan makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi. 9 fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak pesan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, bisa memberi nilai tambah bagi pelaku industri. Kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antar akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar 3 unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Dan inovasi mencabut. 9 fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2024, inovasi berdasarkan Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045 Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi dengan strategi yang dibangun Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan, hingga bagaimana inovasi tersebut bisa memberi nilai tambah bagi pelaku industri. Itulah sebabnya, kemampuan menguasai pasar dan jaringan distribusi merupakan bagian yang sangat penting dalam sebuah proses pengembangan produk inovasi. Sinergi antara akademisi, pebisnis, pemerintah, merupakan bagian penting sebuah strategi hilirisasi inovasi pembangun ekonomi bangsa. Bekerja dalam sinergi Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional adalah kunci utama. Tertuang dalam prioritas riset nasional 2020-2024, inovasi berlandaskan riset yang terfokus dengan kolaborasi antar tiga unsur triple helix dapat mendorong inovasi yang berdaya saing dan membentuk SDM iptek unggul. Karena integrasi riset dan inovasi untuk Indonesia Maju. Mobilitas tinggi, kebutuhan yang meningkat, lahirkan percepatan teknologi untuk kemudahan manusia di setiap detik dan langkah beraktivitas. Tingginya standar kompetensi, kualifikasi, dan kualitas sumber daya manusia membuat penguasaan iptek menjadi kunci utama dalam era percepatan. Visi 2045, Indonesia Berdaulat, Maju, Adil, dan Makmur telah dicanangkan. Investasi dan ekosistem yang mendukung dalam riset dan inovasi adalah kunci untuk mencapai visi tersebut. Sembilan fokus riset telah ditetapkan. 49 produk inovatif telah direncanakan guna mendukung transformasi ekonomi Indonesia from resource-based economy to innovation-based economy. Dengan daya saing manufaktur dan jasa modern mempunyai nilai tambah tinggi bagi kemakmuran bangsa. Inovasi diperlukan agar sebuah bangsa senantiasa berkembang. Perkara inovasi bukan hanya soal hasil riset, melainkan membawanya ke dunia industri. Diperlukan banyak terobosan yang melibatkan banyak pihak, mulai dari bagaimana hasil riset tersebut diwujudkan,
Kenapa, Bu?
Recording in progress.
Good afternoon, ladies and gentlemen. Thank you for joining our seminar. I am Faiza Mariani. I'm moderator for this parallel session. In this parallel session, we will discuss about pharmaceutical technology, drug delivery system, natural product, and medicinal chemistry. We will have our co-host here, who is Dilani Putri, who will help us to play the video presentation of the participant. We will have 8 speakers here who will present their topic. Every speaker will present for 10 minutes. I will divide it. The 8 speaker become 2 groups. The first group is consists of 4 speakers who will present their topic and continue with discussion. And after that, we will continue to another group who there are 4 other speakers who will present their topic and continue with discussion. If you have any question, you could deliver your question to the chat room. or you could use raise hand and active the video and audio to ask question directly. For asking question, you could use Bahasa Indonesia if you want. I will check the list of our speaker here. We have 8 speakers. For the first speaker, we have Mrs. Jamila Abbas. Hello, Mrs. Jamila Abbas. Okay, Mrs. Jamila Abbas is already here. And
yes,
the
I am here.
Oh yeah, okay. The second speaker we have Mrs. Yeni Meliana. Hello, Mrs. Yeni Meliana. Oh, maybe she is not here. Okay, the third speaker we have Eva Maria Widiasari. Mrs. Eva Maria Widiasari.
Yes, I am here.
Okay, thank you. For the fourth speaker, we have Mr. Ahmad Fatoni. Hello, Mr. Ahmad Fatoni.
Hello.
Hello, yeah. Thank you for your presence. And the next speaker, we have Mrs. Sofa Fajria.
Yes, I'm here.
Okay. And the next speaker, we have Mr. Muhammad Eka Prastia. Hello.
Yeah. Hello.
Yeah, okay.
Yes.
And the next speaker, we have Mrs. Teni Ernawati. Hello, Mrs. Teni Ernawati. Oh, she's not here yet. The last speaker, we have Mr. Anjar Purba Asmara.
Yes, I'm in.
Okay, thank you for your presence. Okay, maybe we could start our first presentation here. Will be delivered by Mrs. Jamila Abbas, who will present her topic about chemical constituent, anti-cancer, and antioxidant activities of neonuclear medicinal leaves. Okay, maybe I would like to ask the co-host to play the video presentation, please.
Thank you very much for moderator. In this occasion, I want to explain my research with topics chemical constituent, cytotoxicity, and radical scavenger activity of neonuclear medicinal leaf. Introduction. Indonesia is the number 2 ranked country in biodiversity after Brazil. WHO stated about 20,000 medical plants are used in the pharmaceutical industry. Over the past 50 years, 500,000 existing plant species have been investigated. Only a little has been studied for bioactivity. Therefore, there are great potential for future discovery, new medicine from Indonesian plants. Indonesia until now still import 95% medicine ingredient. Cancer and oxidation case. At 2013, cancer can result in death and mortality died estimate 40,350 and almost 1,600 people died per day. At 2030, estimate cancer will expected to grow up to 21.7, 11 cases, 13 million deaths. Reactive oxygen species or ROS can easily damage the of the cell membrane, that is phosphatidylipid lipoprotein by propagation reaction cycles. It has been mentioned that antioxidant activity of plant is due to phenolic compound. We use N-calicina leaf for curing cancer and oxidation reaction because only a few researchers on the phytochemical analysis and bioactivities of this N-calicina are reported. The effective treatment of the cancer and oxidation will increasingly depend on the active compound in the material plant. N-calicina leaf, according to the family Rubiaceae, have 26 genus and 40 species. The tree have 24-meter tall and 44 centimeter diameter, and the tree will live in mountain up to 1,600 meter altitude among the river, and also will live in hillside and shrub. The tree will distributed in Burma, Indochina, Thailand, Malaysia, Sumatra, Kalimantan, Philippines, and Celebes. One of the benefits of this plant is a wood medicine. The Rubiaceae family are economically important as a source of medicine, also for building and nutrition, and the leaf of the N-calicina were foremost traditional medicine in diet tribes. The Rubiaceae family tree economical important as source of medicine. As anti-DNA topoisomerase effect, the leaves were collected at Kalimantan Center and leaves were dried and extracted with hexane, ethyl acetate, and methanol. The total phenol, total flavonoid, radical scavenging activity, cytotoxicity. In this research, gallic acid, natrium also. Method. Total phenol content was determined by Folin KTQ, and total flavonoid content was investigated by using aluminum chloride funding. We hope our extract have activity as cytotoxic and radical scavenging. Radical scavenging activity were determined by using DPPH. Cytotoxic activity was done by using MTT reagent and breast cancer saline. The result: total phenol content were calculated using the following linear equation based on the calibration curve of gallic acid. And total flavonoid content were calculated using the calibration curve of quercetin. Cytotoxic assay was expressed as IC50 of 3 replicate analysis. MTT assay, the cytotoxicity activity of N-calicina leaf extract again breast cancer saline were analysis with colorimetric MTT assay various concentration of the sample 50 until 500 ppm. We also include untreated cell as control. After 24-hour incubation period, the cell viability can be qualitative assessed by the color intensity of the purple parmesan dye.
Okay, maybe I think there is trouble with the video presentation. Maybe I would like to the co-host to check it. Is it finished or something technical problem?
I think we lost connection for a bit. I will try to share screen again and with the last presentation. Apologize for the technical issue.
Okay, thank you.
Mbak, kayaknya saya ada yang berisik deh, Mbak. Ini saya nggak jalan apa ya? Yang lonceng, Pak. Udah, Pak, tadi udah. Apa ya, Pak? Ini aja.
Nggak bisa lah ngomong.
Berjalan lagi sih, Pak.
Breast cancer cell line analysis with link MTT assay various concentration of the sample 50 until m. We also include the cell viability can be qualitative assessed by the color intensity of the purple pharmazine dye. When the concentration of the extract decreases, the intensity of the purple pharmazine dye increases. Inhibition of the cell viability caused by hexane extract, ethyl acetate extract, and methanol extract from the N-calicina leaf was examined using MTT. The result will be shown in this picture. The morphology on cytotoxic assay, this by untreated sample, this by methanol extract, this by hexane extract, and this by ethyl acetate extract. This table shows that content of phenol, total flavonoid, and cytotoxicity, scavenging activity of hexane extract, ethyl acetate extract, and methanol extract. Total phenol is milligram gallic acid equivalent, time identification extract. There are some compound, for example, there are 3-tertian butyl methoxyphenol and stigmastan 3.6 dion. And structure, the compound in hexane extract, ethyl acetate extract is stigmastan and also viscosine lactam. Structure compound in the methanol extract, 3 compound, carboxythiodiamine, camphorol, and quercetin. Conclusion, the methanol extract of the sample leaf had the best scavenging effect against the PPH. It's suggested that the sample can be used to prevent and control the oxidative stress induced by free radical. The methanol extract of the sample leaf had the less cytotoxicity effect against breast cancer. It is suggested that methanol extract of the sample cannot be used to prevent and control the proliferation cell. The ethyl acetate extract of the sample leaf had the best cytotoxicity effect against breast cancer. It is suggested that ethyl acetate extract of sample can be used to prevent the proliferation cell. Our research provides evidence that hexane and ethyl acetate of the sample can decrease proliferation cell cancer, inhibit oxidation activity in vitro. Conclusion, in the study, we found that ethyl acetate extract of N-calicina possesses cytotoxicity and radical scavenger activity. The result revealed that the both hexane and ethyl acetate extract has significant cytotoxicity and radical scavenging activity against the PVH. The cytotoxicity and radical scavenging content. Therefore, hexane extract and ethyl acetate extract of the sample could be excellent source of natural cytotoxicity and antioxidant agent for medicinal and pharmaceutical application. We thank to Mr. Sebayang from Kuala Kapua Central for Kalimantan for kindly providing the sample. And also thankfully for research grant for DIPA, Deputy Engineer Science, Indonesian Institute of Science. Thank you very much.
Okay, thank you for the presentation. We are so sorry for the technical issue that happened. So we are going to the next speaker, Mrs. Yeni Meliana. I will check if Mrs. Yeni Meliana is already here or not. Hello. Hello, Mrs. Yeni Meliana.
Yes.
Okay, thank you for your presence. Okay, I would like to ask the co-host to present the video presentation from Mrs. Yeni Meliana. Please play the video.
Thank you for the committee. My name is Emiliana and my team from Research Center for Chemistry, National Research and Innovation Agency in the Republic of Indonesia. Here we will share about the lyophilase solid dispersion of artemisinin to improve saturation, solubility, and dissolution. This is for the introduction. Since 19 April 2006 in Geneva, WHO recommend the use of artemisinin-based combination therapies in order to end resistance. For the artemisinin from this Artemisia annua plant, this is a plantation of Artemisia annua in Cibodas, Bogor. We know malaria is still become a global issue, so WHO keeps to try to solve this issue because a lot of drug is become resistant right now. This is from the cultivation from the Artemisia annua plant. Another group in our group research conduct the research for the extraction to get this artemisinin from the Artemisia annua plant. This is a structure molecule of the artemisinin. From this artemisinin, this is water insoluble, so derived to this artemisinin become another structure molecule here. Artemisinin has a poor bioavailability, limiting its effectiveness. Therefore, semi-synthetic derivative of artemisinin, here we can see dihydroartemisinin, this is the structure, artemether, artether, artesunate, and artelinate have been developed. This is from Baikas 2009. So, what we become the issue for this artemisinin? This is artemisinin For this peroxide bridge, this is peroxide bridge, this is peroxide bridge. All these peroxide bridges only have the limit only 40 Celsius degree. Above 40 Celsius degree, this brick will be broke. So anti-malaria properties from this structure molecule will be disappear. Besides that, artemisinin have the properties is hydrophobic or water insoluble. Also low bioavailability and only solve in ethanol or methanol. So it's different with the derivative of this artemisinin. This dehydroartemisinin, this one, dehydroartemisinin. This is have property water soluble. So it's prepared become the anti-malaria drugs. But the problem to get this dehydroartemisinin from artemisinin is quite expensive because it need the expensive catalyst here, and then spend 3 hours and also other chemicals to derive like ethyl acetate and also methanol. So what we will do? Here we investigate the development of solid dispersion artemisinin so that is hydrophobic or water insoluble. We put in solid dispersion using the matrix maltodextrin. This is from polymer, natural polymer maltodextrin and natural gum arabic using the method, the freeze-dried method. This is figure artemisinin, this is figure for the gum arabic, and this one for the maltodextrin. Methodology, first dissolving 57% maltodextrin and gum arabic in distilled water and addition of 3% artemisinin in ethanol. And then homogenize for the 30 minutes and speed of 50,000 RPM. Evaporate the ethanol by using rotary evaporator and then frozen all the mixture at minus 23 Celsius degree. And then freeze dry under vacuum and condense at minus 45 Celsius degree until the sample was completely dry. Experiments were performed in triplet and encapsulation efficiency was calculated using the equation. This is equation for entrapment efficiency in percent. This is result. Same image, show the morphology. This is figure for the artemisinin crystal. This is B, C, and D. When the artemisinin with the solid dispersion with the maltodextrin and gum arabic. For this one, with the ratio between maltodextrin and gum arabic is 50%. Here, when we increase amount of the gum arabic, for the D, when we decrease amount of gum arabic. Calculate using image J application, the article size of artemisinin solid dispersion was in the range 22 till 420 micrometers, while the crystal before solid dispersion is in the size particle 313 till 1223 micrometers. It indicates the solid dispersion of artemisinin in maltodextrin and gum arabic can reduce the particle size. This might affect the dissolution properties since particle size reduction can increase the dissolution rate of the drugs. This is result for the efficiency of entrapment. We can see here when the artemisinin with the solid dispersion with the ratio between maltodextrin and gum arabic is 50%, entrapment efficiency is 88%. When increasing amount of gum arabic, the entrapment efficiency is increased, become 96%. When we reduce amount of gum arabic or more maltodextrin in the solid dispersion formulation, the entrapment efficiency also decreased, becomes 79%. The increasing amount of gum arabic in the formulation was found to increase the entrapment efficiency. The ability of gum arabic to carry out the process of suspending, emulsifying, and stabilizing. Gum arabic can act as stabilizer for oxide dispersion and individual carbon nanotubes in aqueous solution. Here, gum arabic addition will increase entrapment efficiency, but gum arabic has drawback in terms of price. Gum arabic is considered expensive if compared to maltodextrin. Or the maltodextrin has good compatibility with gum arabic, and its addition will reduce the cost. This is data for saturation solubility, solubility of pure artemisinin and artemisinin solid dispersion. This is, we can see solubility if only artemisinin is 47, when the ratio between maltodextrin and gum arabic for this artemisinin in this solid dispersion, the solubility is become 59. When we increase more gum arabic, its solubility value is 60. When we reduce amount of gum arabic and more maltodextrin, yes, the solubility is become 56. The solubility of original artemisinin crystal in water was observed to be 47.68 milligram per milliliter at 25 Celsius degree room temperature, indicate the artemisinin has poor solubility in water. The solubility of artemisinin was increased after dispersed in maltodextrin and gum arabic carriers. The freeze drying method was reported to effectively enhance drug solubility, perhaps due to an increase in surface area and the surface free energy. According to Noyes-Whitney equation, the saturation solubility of the drug can be increased by the particle size reduction in order to increase the particle surface area. This is data for in vitro dissolution study. The dissolution profile of lyophil solid dispersion exhibit an improvement in the dissolution behavior formulated artemisinin compared to pure artemisinin crystal. This is pure artemisinin crystal. This is for the artemisinin solid dispersion. Artemisinin show a slow release profile because of its poor solubility. The dissolution rate of artemisinin crystal was slow, with less than 5% of artemisinin dissolved in 1 hour. This is, we can see, till 1 hour, the dissolution still less than 5%. Even after 3 hours, here, the pure drug did not show 50% dissolution. This might be due to the poor wettability and the agglomeration of particles during the process, so that the powder floated on the surface and dissolution medium. For the conclusion, first, preparation of artemisinin solid dispersion in maltodextrin and gum arabic using the freeze drying methods can increase artemisinin saturation solubility due to the decrease in particle size. Based on entrapment efficiency and saturation solubility data, increasing gum arabic in artemisinin solid dispersion would increase the entrapment efficiency and solubility. Gum arabic has the ability to suspending and stabilizing the mixtures and giving more interaction with artemisinin. And in the dissolution study, the lyophils artemisinin solid dispersion exhibit higher drug release due to the interaction of drug with the carriers. The hydrophilic effect of maltodextrin and gum arabic increase the dissolution because they reduce the hydrophobicity of artemisinin and the interfacial tension between artemisinin and the dissolution media. Thank you.
Thank you so much for your presentation. We are going to the next speaker, who is Mrs. Eva Maria Widyasari. She will present her topic about the physical-chemical characteristic of 99-MT-CD-TPA deoxyD-glucose as radiopharmaceutical for cancer diagnosis. I would like to ask the co-host to play the video, please.
Assalamualaikum warahmatullahi wabarakatuh. Today I will present my research with the title Physical-Chemical Characteristic of 99M Technetium DTPA deoxyglucose radiopharmaceutical. And this research team involved in this study are me, Eva Maria Widyasari, Aulia, Witri Nuraini, Teguh Hafiz, and Maula Eka Sriani. As we all know, that cancer cases are increasing from year to year while there are still few methods to early detect the presence of cancer in the body. The cause of cancer is not clearly known, but this is in line with the change of the lifestyle of modern society and pollution caused by polluted environment. Data in 2018 show that cancer cases are 18.1 million and cases of death from cancer are about 9.6 million. Radiopharmaceutical are unique medicinal formulation containing radioisotope, which are used in major clinical area for diagnosis or therapy. Radiopharmaceutical generally consists of two components, a radioactive element that permit external scan and a non-radioactive element that responsible for conducting the radionuclide to a specific organ. In nuclear medicine, radiopharmaceutical are non-invasive visualization that used in diagnostic imaging and therapy. In the imaging modality, radiopharmaceutical are administered via oral, intravenous, or by inhalation to enable visualization with their radioactive tracer of various organs. Deoxydiglucose is glucose molecule which has the hydroxyl group replaced by hydrogen, so that it cannot undergo further glycolysis and it acts to competitively inhibit the production of glucose 6-phosphate from glucose at the phosphoglucosimerase level. Deoxyglucose is uptaken by the glucose transporter of the cell. Therefore, cell will higher glucose uptake. For example, tumor cell has also a higher uptake of deoxyglucose. Therefore, 2-deoxyglucose is very potential to be used as a radiopharmaceutical. Therefore, in this study, the radiopharmaceutical 99M DTPA deoxyglucose was developed. In the manufacture of radiopharmaceutical, there are 4 stages of activity that must be carried out. There are the labeling process, determining the physical-chemical characteristic, preclinical testing, and the clinical trial of radiopharmaceutical. And in this research, we will report the physical-chemical characteristic of the radiopharmaceutical 99M technetium DTPA deoxyglucose. 6 physical-chemical characteristics that analysis were visual. This is done by observing the resulting formulation using a dark background to determine whether the resulting preparation is cloudy or discolored. Radiochemical purity. Radiochemical purity, this is done by paper chromatography, which can separate radiochemical impurity. Lipophilicity. This is done by measuring the value of the partition coefficient in mixture of organic solvent and water. Electric charge. This is done by electrophoresis method. Plasma binding. This is done by determine the concentration of radiopharmaceutical that are free in plasma or bound with plasma protein, and this is done by separating the supernatant and precipitate using centrifuge. And the last is stability. This is done by determining the radiochemical purity of radiopharmaceutical that have been stored for a certain period of time. The results obtained from this study are from the visual test. This is known that a radiopharmaceutical formulation look clear and colorless. This is so that 19N technetium DTPA deoxyglucose is visually safe to use on injection preparation. Radiochemical purity. From several repetitions, the radiochemical purity of 19N technetium DTPA was 95.2%. The radiochemical purity result of 19N technetium DTPA meet the requirement according the two British Pharmacopoeia and the United States of Pharmacopoeia because the radiopharmaceutical purity percentage is more than 90%. From 3 repetitions, the lipophilicity of 19N technetium DTPA deoxyglucose was -1.2, which means it is hydrophile. This show that 19N technetium DTPA deoxyglucose will be excreted through the kidney, so the radiopharmaceutical is not toxic and its radiation effect on body organ is relatively low because it will quickly excrete through the kidney. Electric charge. This is important to determine because the interaction between the radiopharmaceutical molecule and the surface of the body cell membrane depend on the electric charge of the molecule and the permeability of the membrane. From this figure show that 19N technetium DTPA deoxyglucose stay at 0 point, mean has a neutral charge. Plasma binding. The plasma binding is determined the concentration of radiopharmaceutical that are free in plasma or bound with plasma protein. The value of the bond will affect the coefficient of the use of the drug. The results show that 19N technetium DTPA deoxyglucose had a degree of binding to plasma protein of 72.0%. This will affect the length of the biological half-life of the radiopharmaceutical. This giving the effect of sufficient time for imaging with a gamma camera in nuclear medicine. And the last, stability. Determine the stability of the deoxyglucose liquid kit in storage which aim to determine shelf life. The stability test of the deoxyglucose liquid kit was carried out for up to 60 days, which was observed every week by labeling with technetium 99M, and the determining the radiochemical purity using chromatography. Until 6 days of observation, the quality of the deoxyglucose liquid kit is still good, with the radiochemical purity above 95%. And the conclusion from the study was obtained physicochemical characteristics that can use as reference in a subsequent in vivo study of 19N technetium DTPA deoxyglucose radiopharmaceutical. From in vivo study, we'll get a comprehensive information on the bioactivity of this compound for diagnostic cancer. Thank you.
Okay, thank you for your interesting presentation. Okay, next we will have Mr. Ahmad Fatoni. He will present the topic about evaluation of hypoglycemic effect of 1.1 bislunatin in streptozotocin-induced DDY, male mice, or mus musculus. I would like to co-host to play the video, please.
Good afternoon, honorable ladies and gentlemen. Thank you for your time. My name is Ahmad Fatani. I come from Research Center for Chemistry. In this occasion, I will discuss and share related to my research entitled Evaluation of Hypoglycemic Effect of +1,1 Bislunatin in Cytosine-induced DDY male mice, musculus. This is the outline of my presentation. As we know, diabetes mellitus is a metabolic disorder that tends to cause hyperglycemia due to impaired glucose tolerance. Impaired glucose tolerance due to several conditions such as increased gluconeogenesis in liver, insulin resistance, increases lipolysis, and beta cell compensation and decompensation. The insulin resistance due to obesity, genes, aging, and oxidative. Based on WHO, there are three main types of diabetes, namely type 1 diabetes, if the body does not produce enough insulin, type 2 diabetes if the body produces insulin but cannot use it well, and the third one is gestational diabetes, a temporary condition in pregnancy. Based on WHO, in 2014, there are 422 million adults have diabetes, or 1% in 11% have diabetes, and 3.7 million deaths due to diabetes and high blood glucose, and 1.5 million deaths caused by diabetes. Until 2013, in the worldwide, almost 400 million people have been suffering from diabetes, and it is projected that the number may increase to nearly 600 million by 2035. One of the main goals of medical therapy for diabetes mellitus type 2 is to control blood glucose level by administering oral hypoglycemic drugs. However, in practice, oral hypoglycemic drugs limited efficacy and have adverse side effects such as hypoglycemic and lactic acidosis. This is the reason has led to increasing interest in the use of natural resources derived from plants or endophytic fungi. Hypoglycemic activity as alternative management in treating diabetes mellitus patients. Skyrin, as we know, is a bisanthraquinone produced by the fungus Saccharomyces fortmani, has been previously reported as a glucagon antagonist and can reduce blood glucose level. We can see the chemical structure of BSL and skyrin. BSL have a chemical structure that is identical to skyrin and differs only the methyl group at the C number 7 position. And this is BSL have 2 methoxy groups and skyrin have 2 methyl groups. Therefore, based on the structure-activity relationship rule, it is assumed that bislunatin also have similar activity to skyrin as a glucagon antagonist against type 2 diabetes. Based on this presumption, it is necessary to evaluate the anti-diabetic activity of bislunatin. This is the compound of bislunatin powder. Before we use animal testing, the animal were acclimatized for 1 week to adapt to their environment, and the 30 male mice were randomly divided into 6 groups, each consisting of 5 mice. This is the flowchart of experimental study. You can see there are 6 groups of animal testing, namely normal, negative, positive, low dose, moderate dose, and high dose of bislunatin treatment. Blood glucose level measure were carried out 3 times, namely before cytosine induction as baseline, after induction, and after treatment. Blood glucose, this is the result of this study. We can see blood glucose level as baseline before cytosine induction is 150.40 milligram per deciliter. This study showed the citizen induction caused hyperglycemia in the red box. Blood glucose level is above 250 milligram per deciliter, or considered as a diabetic mice. Based on the result of the blood glucose level value in the post-induction for normal control compared to all groups with cytosine induction, streptozotocin induction is significantly different. The result of blood glucose level in the post-treatment show that the normal control group was not significantly different from the positive control group and all those of the bisglutamine treatment group. This data show that the all treatment have blood glucose level under 250 milligrams or non-diabetic mice as well as normal groups. Meanwhile, the negative control group with streptozotocin induction group and without treatment metformin have blood glucose level above 250 milligrams or still considered as a diabetic condition. This is the percentage of increased blood glucose level of the group with streptozotocin induction. Negative group is without treatment, and we can see, however, all data have percentage decrease. Glucose level is not significantly different, but the negative control still considered diabetic. Their blood glucose level is above 250 milligram per deciliter, and all treatment, both positive and all BSL treatment, have the criteria of mice of blood glucose level after treatment are non-diabetic mice. Another study stated that there was no significant difference in the decrease in the blood glucose experimental animals with increased dose. Although the highest dose, there was the greatest decrease in the blood glucose level. Oral absorption of drugs is very complex system. There will occur interaction between physiology and physicochemical factors and metabolism. The effect of bisglutamine, which can lower blood glucose level, is expected to be a candidate for type 2 diabetes. In this study, metformin was used as comparison drug in positive control group. Metformin is one of the biguanide oral hypoglycemic drug that work directly on the liver, cause a decrease in hepatic glucose production without stimulating insulin secretion. However, the side effect of using metformin can cause depression of lactic acidosis. Lactic acidosis occurs in the presence of buildup of lactate caused by an increase in lactate production that results in hypoxia or decrease in lactate elimination. The advantage of use BSL is glutamine, which can decrease blood glucose level in this study, as well as have no serious side effect with oral low acute toxicity based on previous study. So bisglutamine may be developed into an option to treat diabetes mellitus in the future. In the conclusion, all mice after induction by streptozotocin show considering diabetic mice criteria. All treatment with several doses of bisglutamine and positive control would reduce blood glucose level and make it lower than 250 milligram per deciliter, as well as blood glucose level of normal control. The percentage of decreased blood glucose level with the treatment of several doses of bisglutamine was not significantly different from the positive control. The percentage of decreased blood glucose levels with treatment bisglutamine is higher than without treatment. Thank you very much for your attention.
Okay, thank you so much for your nice presentation. So we are going to do the discussion part. If you have any question, you could deliver your question to the chat room or raise your hand and active your video and audio to ask question directly. Is there any question from the participant? Okay, maybe I would like to ask a question for Mrs. Yeni Meliana. You said that you make the lyophilized solid dispersion, and I want to know that how you check the. Okay, you have solid dispersion, and that how you check the release rate or entrapment efficiency to get the number. How you check in the lab, the procedure or the method. Did you use dialysis method or something like that? Thank you.
Okay, thank you. Thank you, Ms. Faiza, for the question. Yes, in our work, we try to make a solid dispersion for artemisinin, which is a hydrophobic compound. We put it together with the matrix polymers, gamma arabic and maltodextrin, and then we try to measure dissolution. Your question is dissolution, right?
Yeah, the release and how to check the entrapment efficiency and how to check the release rate of your sample. Yeah.
Yeah. To check the release sample, we use the UV spectrometer, and then we calculate by the equation entrapment efficiency is a ratio between experimental drug loading compared to nominal drug loading. So that's the way how we.
Did you check the how to say? If you use some drug in the body, you have different pH level, something like that, and that. Did you check different pH environment from the sample, or just one neutral or something like that?
Oh yeah. We actually measure in simulation 3 kind of the simulation when measure it's in the mouth is with the, so we measure in the 6.8 pH, 2.7 pH, and the other one is 4 pH, if I'm not mistaken. Yeah, in 3 kind of simulation, just like you said.
Oh yeah. Which one is the best one? Which pH?
Which one the best? Okay, I will check. The best one is when it's in the 6.7. Actually it's 6.7. Actually we the best calculate the drug release is when it's also good when it's released at the pH 2.4, right? When it's in the stomach, colon stomach.
Yeah. But for your case, it's better when 6.8.
Yeah, in normal pH.
Okay. Thank you so much for your answer.
Thank you, Ms. Faiza.
Yeah, we have a question from Miss Tenny from Mrs. Tenny to Mrs. Eva Maria. The question is how to choose the right radio label for drug candidate compounds. Maybe Mrs. Eva could answer the question, please.
Okay, thank you for the question. I will try to answer in Indonesia. This is 2 questions, right? And 2 questions.
Oh yeah, 2 questions. The first I said that from Mrs. Tenny, and the second one from Mr. Eka to Mrs. Eva. Is this the question is
okay.
Yeah.
Okay. Baik.
Thank you.
Saya coba menjelaskan dalam bahasa Indonesia karena biar lebih jelas gitu ya. Jadi untuk suatu sedia radiofarmaka itu pertama kali yang kita lakukan adalah menguji
karakteristik fisikokimianya gitu sebelum dia digunakan ke preklinis. Jadi tahapannya setelah radiofarmaka itu jadi, kemudian kita uji preklinis dulu baru klinis seperti itu kan ya. Nah, ini tahapan yang saya laporkan dalam riset ini adalah preklinis pengujian fisikokimianya gitu sebelum ke preklinis. Nah, untuk sediaan radiofarmaka itu kan biasanya salah satunya kita menggunakan cara intravena gitu ya. Untuk digunakan secara intravena itu syaratnya kan ada beberapa macam. Misalnya sediaannya harus jernih, dia tidak toksik seperti itu. Nah, di sini yang saya laporkan tadi di sini kan yang ditanyakan adalah secara visual gitu ya. Secara visual tadi saya laporkan bahwa sediaan itu jernih dan tidak tidak keruh seperti itu. Jadi secara visual ini bisa digunakan aman digunakan sebagai sediaan radiofarmaka yang diberikan secara injeksi. Cuman bukan hanya pengujian itu saja sebenarnya, nanti toksisitasnya seperti apa, pirogenitasnya seperti apa itu juga harus dicek gitu. Nah, kemudian pertanyaan yang kedua di sini tentang tentang bagaimana cara memilih senyawa yang bisa dilabel dengan baik gitu ya, kandidat. Nah, jadi sebenarnya untuk senyawa itu sebenarnya ketika kita mau menandai suatu senyawa, itu kita lihat strukturnya seperti apa, kira-kira yang cocok untuk suatu, misalnya kita punya senyawa A gitu ya. Nah, kira-kira nanti isotopnya apa yang sesuai, yang cocok untuk menandai senyawa itu. Jadi biasanya bisa menggunakan yodium itu berarti nanti substitusi elektrofil misalnya seperti itu, atau reaksi adisi seperti itu. Jadi biasanya kita pelajari dulu struktur kimianya seperti apa, baru kita menentukan isotopnya. Kemudian dari situ kita pelajari secara preklinisnya apakah benar senyawa itu mekanismenya sampai ke target organnya, organ targetnya. Misalnya untuk kanker di sini yang saya bahas ini untuk kanker, nanti kedepannya akan kami coba ke hewan model kanker, apakah benar senyawa tadi itu akan terakumulasi banyak di kanker, misalnya seperti itu, sehingga dia bisa dijadikan radiofarmaka untuk diagnostik kanker gitu. Memang belum sampai ke preklinis untuk saat ini. Itu mungkin untuk jawaban yang kedua. Thank you.
Is there any comment or any opinion from the people who asked the question? Okay, I think it is all clear, and we are going. Okay, thank you for your answer. For the next question, we got from Mrs. Rizna to Mr. Fatoni.
Oke, saya bahas dalam bahasa Indonesia terkait dengan farmakodinamik tes terhadap bislunatin yang dapat menurunkan kadar gula darah, apakah dibandingkan dengan metformin sebagai kontrol positif. Ini bisa dijelaskan? Oke, untuk saat ini yang telah dilakukan untuk tes uji in vivo terhadap bislunatin maupun metformin ini menggunakan mencit itu terkait dengan kadar gula darah setelah diinduksi dengan menggunakan streptozotocin. Namun terkait dengan uji farmakodinamik belum dilakukan atau belum dilakukan untuk saat ini. Mungkin ini bisa sebagai masukan. Namun terkait dengan efek side effect atau efek samping, kemungkinan akan berbeda ya, karena dari terkait dengan cara kerja dari metformin maupun bislunatin itu sendiri. Secara literatur kita bisa melihat kalau bislunatin dari strukturnya itu mirip dengan skyrin, dan dia akan menginhibisi terkait dengan enzim yang ada di liver, dan akan sedikit berbeda dengan metformin seperti itu. Kalau kita perhatikan dari studi literatur seperti itu, Ibu. Terima kasih.
Oke, terima kasih. Thank you for your answer. Okay, I think our discussion part is finished now, and we would like to go to the next speaker. The second group we have Miss Sofa Fajria, Mr. Muhammad Eka Prastia, Mrs. Teni Ernawati, and Mr. Anjar Purba Asmara. Okay, for the first speaker in the second group, I would like to introduce Ms. Safa Fajria, who will present her topic about bioassay guided isolation of Indonesian sea with anti-diabetic activity from the Artocarpus elasticus ex Bloomberg. So I would like to ask the co-host to play the video, please. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like thanks to the organizing committee for giving me opportunity to give the presentation about bioassay guided isolation of Indonesian sea with anti-diabetic activity from Artocarpus elasticus bark. The outline will be delivered about the introduction, materials and methods, results and discussion, and conclusion. As we know that diabetes mellitus is a metabolic disorder characterized by hyperglycemia associated with abnormalities of carbohydrate, fat, and protein metabolism caused by decreased insulin secretion or decreased insulin sensitivity, or both, and cause chronic and neuropathic complication. The cause of diabetes mellitus is lack of hormone insulin. So as a result, the glucose builds up in the blood and eventually extracted in the urine without being used. And another cause is decreased sensitivity of cell receptors to insulin and caused by overeating and obesity. Some herbal medicine have been reported to be useful to treat diabetes and have been used empirically as diabetic remedies. One of plant families known to have many benefits is Moraceae, and one of the genera belonging to the family Moraceae is Artocarpus. Artocarpus is widely distributed throughout the archipelago, and several species are endemic to South Sulawesi. And this Artocarpus, known to contain plant-related phenolic compounds, especially flavonoids with various structural variations. And the prenyl group on the flavonoid is in the C3 position and deoxygenated in bearing at the C4, C2, C4 accent, C2 accent, C4 accent, or C5 accent position. And in addition, prenylation can also occur at the position C6, C8, and C3 accents. Artocarpus elasticus also has a unique structures of flavonoids and produce broad psychological effects such as artonin E, artobilosanton, psychoartobilosanton, artocenon P, and artoflavin B as alpha-glucosidase inhibitors. So, the purpose of this study was to isolate alpha-glucosidase inhibitory compound from Artocarpus elasticus bark using bioassay-guided fractionation techniques and identify the isolate using spectroscopic methods. The materials and method we use the bark of Artocarpus elasticus collected from Mekonga Forest, Southeast Sulawesi, Indonesia, and identified by botanists from the Center for Biology LIPI. Now is brain. A voucher specimen was deposited in the herbarium with voucher specimen UHA 46. After that, we go into the extraction and fractionation, isolation and purification, and alpha-glucosidase inhibitor activity assay. The extraction and fractionation process, first about 800 grams of dry bark of Artocarpus elasticus we extracted with methanol, and the filtrate we evaporated and we got the methanol extracts. After that, the methanol extract we going to the alpha-glucosidase inhibitor activity assay, and then we fractionated of the methanol extracts using normal hexane, ethyl acetate, and butanol as the solvent, and we got the normal hexane fractions, ethyl acetate fraction, butanol fraction, and water as residue. And all these fractions we examine the alpha-glucosidase inhibitor activity assay. The ethyl acetate fraction, we going to the purification with column chromatography with gradient elution, normal hexane, ethyl acetate, and methanol, and we got 9 fractions. And after that, we examine the alpha-glucosidase inhibitory activity assay. The subfraction 9 is the highest activity against alpha-glucosidase enzyme, and we go after that we purification using Cepadex LH20 with MTC and methanol as the solvent, and after that we going to the preparative TLC, and we got we got the pure compound as compound 1, a yellow powder, and we identify using UV-Vis, FTIR, LCMS, and FTNMR. For the alpha-glucosidase inhibitory activity assay, first we add the sample with the phosphate buffer and PNPG, and we incubated 5 minutes. After that, we added with enzyme alpha-glucosidase solution, and then we incubated 15 minutes, and the reaction we stop with natrium carbonates. The blanks we prepare to correct background absorbance where the enzyme was replaced with 250 microliter of phosphate buffer, and we got the percent inhibition of alpha-glucose inhibitory activity, and the value of IC50 we have from the calculation of alpha-glucose from the inhibitor values by applying linear regression analysis, and we use quercetin and alpha-monglicin as a reference standard. Results and discussions. The extraction process of the dried barks of Althocarpus elasticus yielded about 5% of methanols, and then the extracts exhibit high activity as alpha-glucose inhibitor with an IC50 1.95 microgram per milli. After that, the fractionation process, the yield of the ethyl acetate fraction has the highest yield. So we going to the purification, and then we got the SF5 has the highest in alpha-glucose inhibitory activity with the IC50 1.38 microgram per milliliter. After that, we going to the identification of compound 1. The UV-Vis spectrometry analysis of compound 1 show that there are 2 absorption bands of the maximum wavelength of the 265 nanometer and 395 nanometer. It was characterized for xanthan derivatives. The FTIR spectrum also showed the typical of hydroxyl, aliphatic, carbonyl, and benzene group at absorption. We can see in the we can see from the picture. The analysis of NMR, we can see the proton NMR, carbon NMR, and DEPT NMR. From this spectrum, we can see show the presence of the intermolecular hydroxyl group at the chemical shift about the 12 ppm, and also the presence of 2 methyl groups in here, and then we can see the 2 allophenic protons and isoprenyl groups. From the carbon spectrum, we can see that we have 4 methyl, 4 methane, 2 methylene, and 13 quaternary carbon and 3 carbonyl. So we're going to the HMQC spectroscopy. We can see the correlation between proton and carbon directly. We can see in here that the proton in the 5.50 have correlated with the carbon in chemical shift 28.7, and we can see in here the correlation HMQC of the isolate A. We also use the HMBC correlation to know the correlation between the neck bore until 3 neck bore. We can see in the spectrum And in here we have a correlation between ring A, ring B, ring C, and ring D. And we have the from the literature. From the literature, we can see that the isolate A has similar compound with the artosynin C. So, as a compound 1, we can conclude that the structure is artosynin C. We isolate from Artocarpus elasticus bark. The alpha glucosidase inhibitor activity assay, we can see in here that the artosynin C have the IC50 about 31.88 microgram per milliliter, with the alpha mangostin as the positive control, but with the IC50 about the 14.55. And from this result, we can see that the isolate one has the activity lower than the extract or this fraction. Compared to between the compounds in the fraction level, therefore the artoinosinin C, which is a pure compound, has a lower ability to inhibit alpha glucosidase enzyme. The conclusions: the present study revealed that the artosynin C has been successfully isolated from Artocarpus elasticus bark and potential to be developed as an antibiotic agents. And the extract and fraction from Artocarpus elasticus also have potential source as herbal medicine for antibiotic type remedies. That's all my presentation. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for your nice presentation, Mrs. Sofa Fajriya. So we are going to the next speaker. We will have Mr. Muhammad Eka Prastia, who will present about antimicrobial and antibiovirus activities derived from Indonesian tuna ciliata leaves extract. So I would like to ask the co-host to play the video, please.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for this opportunity. First of all, let me introduce myself. My name is Eka Prastia. I'm a researcher at the Research Center for Chemistry in Indonesia. Well, today I will present our article with the title of Antimicrobial and Antibiofilm Activities derived from Indonesian tuna ciliata leaf extract. For introduction, the overuse of antibiotics in various sectors, in example, public health, agriculture, livestock, etc., it can be modulating natural system mechanism in microbial. At the severe condition, it will creating superbugs microbial, and it will produce or make resistant to antibiotics. At the same time, the microbial biofilm, the condition that one and other microbial attach and produce the biofilm matrix, this phenomena will make it difficult to eradicate by existing antibiotics. So it is required alternative antibiotics compound. On the other hand, Indonesian plant is popular with can produce numerous active compounds, and each of compounds can use for pharmaceutical activities. For example, antioxidant, antimicrobial, anti-cancer, anti-inflammation, and others. And in this study, we investigated 11 Malaceae family plants collecting from Bogor and Bali Botanical Garden. It is including Aglaya volata, Aglaya tomatosa, Eximia, Tixolisum, Apanamyces, Cipadesa, Tona sureni, Tona ciliata, and Kecapi. And overall, the aim of this study is to investigate the potential of fertile extracts derived from several Malaceae plant family to inhibit the growth of some microbial and the biofilm formation. As for the methods, first of all, we have conducted the extraction process from each sample, from dry sample, and conducted the maceration and evaporation, finally get the growth extract. And then for screening antimicrobial activity from each extract, we use 7 microbial tested, including Escherichia coli, Bacillus subtilis, S. aureus, Micrococcus, Pseudomonas aeruginosa, Mycobacter stigmatis, and Candida albicans. 8 of bacteria were cultured in liquid and set to optimal density 0.05. And then each of these culture were applied to 96 well culture site, and also the extract also applied at the same well. After the incubated for 1 night or 24 hours, we observed and the plate zone indicated the concentration of the extract with the microbial growth. The result of this experiment, we obtained that the extract with the code of number 12, it is tonacillata extracts, can inhibit the growth of all tested bacteria with the wide range of antibiotic activity, concentration between 125 until 1000 microgram per milli. Furthermore, we focus to analyze the tonacillata extract, and also only focus with use 3 bacteria tested, including Escherichia coli, Bacillus subtilis, and S. aureus. Each of these bacteria were cultured in liquid and set to microvalent 0.5 and applied to 96 well culture. At the same time, we also applied the tonacillata extract with the different concentration, then incubated for 24 hours. And finally, we observed after the incubation for determining the MIC, it is the level of concentration of the clear zone indicated the MIC value. And also we determine the MPC or minimum bacterial concentration value with the indicating that the concentration with no bacterial colony indicated its MPC value. The result for the experiment indicated that for MIC value, tonacillata extract have the same value with the positive control, it is ampicillin, which show or mention with the orange color in MIC value, and the value is similar. It is 62.5 microgram per milli. It is indicated that tonacillata extract potentially to be developed as antibacterial drugs candidate. However, for MIC value, it is relatively have lowest activity than positive control. Furthermore, we assign for inhibiting the biofilm formation, still focus to tonacillata extract and using 3 bacteria tested. Each of bacteria tested were cultured in liquid culture and set to microvalent 0.5 and applied to 96 well culture. At the same time, the tonacillata extract was applied in this well. After incubated for 24 hours, this experiment was with PBS and treated with 0.5% crystal failure to stain the biofilm. And finally, we detect the biofilm formation with the ELISA reader, and the percentage was measured. The result indicated that tonacillata extract inhibit the biofilm formation at the best activity against Bacillus subtilis. It is mentioned at the yellow graph that it will show that tonacillata extract has the activity against Bacillus subtilis biofilm with the value of 55%, and it belongs to good activity due to more than 50%, and therefore this extract is potentially to be developed as antibiofilm drugs candidate. And finally, for conclusion, tonacillata extract show antimicrobial activity against 7 common microbial, including Bacillus subtilis, S. aureus, and others. And interestingly, the corresponding extract also could inhibit the biofilm formation at the best activity against Bacillus subtilis biofilm. As for developments, this was supported by Grant of Research Collaborative Program between Research Center for Chemistry and Leibniz University of Germany. That's all of my presentation, and thank you.
Okay, thank you for your nice presentation. I would like to remind you we have the attendance form. Maybe you could fulfill the attendance form in the chat room. Okay, for the next speaker, we will have Mrs. Teni Arnawati, who will present her topic about virtual screening approach for an anti-osteoporosis potential from Morinda citriolia fruit extracts. I would like to ask the co-host to play the video, please.
From medicinal research, thank you for taking time for me to present my project research. It's a molecular of the background of this research is the use of Indonesian natural resources for drug discovery development, and also the fact that 1 in 3 women over the age of 50 has osteoporosis. 1 in 5 men over the age of 50 has osteoporosis. There are 200 million people with the osteoporosis in the world and counting. 2 out of 5 Indonesians are at risk of developing osteoporosis. The prevalence of osteoporosis in Indonesia is 42.5%, 2 out of 5 people. Side effect: hypersensitivity, muscle aches, and hypocalcemia. Herbal plants availability and easy to obtain has been proven empirically. Weaknesses of health only empirically. Lack of scientifically literacy. The virtual screening of the chemical constituent from Morinda citrifolia fruit extract for anti-osteoporosis potential through molecular docking approach. The chemical constituent Morinda citrifolia fruit extract, there are beta-diglucopyranose pentacetate to seek the ortho-beta-diglucopyranose 1-octanoyl beta-diglucopyranose, seek O beta-diglucopyranose 1-octanoyl beta-glucopyranose, ascorbic acid, asparolicidic acid, asperucide tetraacetate, scopoletin, isoscopoletin, quercetin, 3O L-ramnopyranosyl beta-diglucopyranoside, caproic acid, caprylic acid, ethyl caprylate, hexanoic acid, and octanoic acid. The material and method. The 2D structure of the active compound Morinda citrifolia was made using ChemDAO11. They are converted into 3D and optimized using the ChemDAO11 application to form MOL2. The minimized of structure MOL2 was using Chem3D. The structure of the osteoporosis protein was downloaded from the protein data bank with the ID number PDB-ID 1TU6. Molecular docking was done using Autodoc 4.2, and visualization of interaction ligand and receptor was used Discovery Studio. The result and discussion. Haptisen key is a lysosome cysteine protease. HAT-key has high collagenation activity. Several papers have shown that HAT-key is the main enzyme involved in the osteoclastic bone resorption and makes it an important target for treatment osteoporosis. In this molecular docking approach, we use the X-ray crystal structure of haptisen key with PDB code 1TU6. HAT-key is an enzyme that plays an important role in the process of osteoclastic bone resorption, and also HAT-key is one of the important targets for osteoporosis treatment. 1TU6 is a haptisen key complex with a ketoamide inhibitor. The slide shows the structure of the chemical constituent Morinda citrifolia fruit extract. This is the chemical structure of beta-glucopyranose pentacetate. This is a chemical structure of 2,6-D or beta-glucopyranosal 1-octanoyl beta-diglucopyranoside. This is a chemical structure of 6 beta-diglucopyranose 1-octanoyl beta-diglucopyranose. This is a structure of asparagolic acid. This is a chemical structure of ascorbic acid. This is structure of asparagolide tetraacetate. This is a chemical structure of scopolamine. This is a chemical structure of scopolamine. This is a quercetin. This is a chemical structure of quercetin, 3-O-ramponitrionyl beta-diglucopyranose. This is caproic acid. This is a caprylic acid, ethyl caprylate, hexanoic acid, octanoic acid, and this is a control positive risodronate for the control positive of molecular docking. This slide shows the result of molecular docking structure of chemical compound contained in Morinda citrifolia fruit extract. In table 1 was described that are 4 compounds out of 15 chemical compounds that are attached to the protein 1-TU6, which have potential activity in inhibiting the activity of osteoporosis. The structural conformation of molecular docking result with the lowest bond energy value for the active compound Morinda citrifolia was shown by the compound quercetin, asparagolide tetraacetate, isoscopolamine, and scopolamine, with energy value respectively -5.77, -5.41, -5.27, and -5.15 kilocal per mole. The positive control of risodonate gives energy value of -3.51 kilocal per mole. This value shows that through a molecular docking approach, the 4 compounds contained in the Morinda citrifolia fruit extract have stronger activity than the positive control. The positive control risodonate has been widely used as treatment for osteoporosis. In this slide, we saw the interaction between ligand and receptor. The best ligand for molecular docking are determined by evaluating their ligand-receptor interaction as well as the energy for osteoporosis ligand-receptor complex being studied. From finding, we found that Morinda citrifolia might be a potential herbal medicine for osteoporosis. We found that quercetin, asparagolide tetraacetate, isoscopolamine, and scopolamine had the lowest binding energy that may result in promising activity osteoporosis decrease. The slide was described the best 3D interaction of binding mode of chemical constituent of Morinda citrifolia extract with 1-TU6 protein. In this slide, we can see the active compound from Morinda citrifolia, such as asparagolide tetraacetate, scopolamine, isoscopolamine, and quercetin in the active pocket site of the osteoporosis protein. And also, in the slide, we can see the control positive risodonate in the active pocket site of the osteoporosis protein 2. The conclusion of this paper are Morinda citrifolia TU6. This protein was selected to blast proliferation. The result of the molecular docking simulation showed that the active compound quercetin, asparagolide tetraacetate, isoscopolamine, and scopolamine have strong binding efficiency against osteoporosis protein 1-TU6 compared to the positive control risodonate. However, further research in experimental, in vitro, and in vivo are needed to see the relationship between the dose of active compound, the response, and the structure of the activity relationship among different proteins responsible for osteoporosis. Financial support from Ministry of Research and Technology, National Research and Innovation Agency, BRIN, Program for Individual Pertama Research in SINAS number 15 A1 KPT 2021. This is a reference that I set for writing this paper. Thank you very much for listening to my presentation about molecular docking of chemical constituent of Morinda citrifolia fruit extract as anti-osteoporosis potential. Hope it can be useful. Thank you very much.
Okay, thank you so much for your nice presentation, Mrs. Feni Arnawati. Okay, we are going to go to the next speaker, who is Mr. Anjar Purba Asmara. He will present the topic about antioxidant potential of ethanolic extract of Sargassum polycystum Ca agar, naturally growing in the large seashore. I would like to ask the co-host to play the video, please.
Hi everyone, I'm glad to be here to present our study about potential of ethanolic extract of Sargassum polycystum, naturally growing in Lange Seashore, Aceh, Indonesia. I'm the first author of this study from Mandiri, Banda Aceh. So this presentation will deliver in 4 sections, open with introduction and then the method, and continue with result and discussion, and then we'll end up with the conclusion. Let's start with the first section of this presentation. Our main concern is oxidative stress, characterized by imbalanced number of pro-oxidants compared with antioxidants. This condition will lead to the development of some metabolic disorders which are fatal for our health and our lives. Some studies suggest that consumption of natural products supplement can be used to combat this condition. One of the example of natural products is coming from Sargassum polycystum, which is a brown cyst wheat that contain a considerable number of bioactives such as phenolic compound, essential oils, and also alginates. This plant is also affordable for those who live in surrounding a maritime area such as Banda Aceh and Aceh Besar. And also this plant has a lower side effects compared with synthetic drugs. And here we will point out our previous studies finding that Sargassum polycystum in the Langi Beach in Aceh Besar has high nutrition and also from and this species could be a source of active phytochemicals that may be beneficial to give antioxidant activities. And also this plant may has a phenolic compound that need to be confirmed in this study. And then let's move on to the method section. So we have 3 main steps. First is a preparation of ethanolic extract of Sargassum polycystum. We call it EES. So we did maceration for 72 hours and we got dry EES about 4.2%. And then we continue the second step by conducting antioxidant assay with DPPH method. We mix our extract with the free radicals and incubate for half an hour, and then we record the optical density to get information about the inhibition percentage. And then we continue with estimation of phenolic content with Foligno-Calcio reagent, and we use the standard with gallic acid, and then we screen the functional groups related to the compounds by conducting infrared spectroscopy. And next, time for the result and discussion. As you can see here in the figure 1, we got vitamin C in the white bar and a brown bar as our EES. From here, from the a lower concentration, we got a percentage of inhibition of our ES surpass 50% of inhibition. So from here, we may say that these extracts could be considered as the active extracts as antioxidant. And when we calculate the IC50, we got lower number, about 2.178 microgram per milliliter. And then we look at the phenolic content, we got 18.572 milligram gallic acid equivalent per gram. And to confirm our finding, we then make a comparison with other studies which conducted to the same species. So here for the figure 2, we can see 7 bar starting from the bottom. The green bar is representing for our study. And then when we compare with 3 studies from Sanur Beach, Kemang Beach, and also Tunggal Gunung Kidul, we got that our values is much higher than those reported from the 3 studies. And then our number also has a similar values from Tamil Nadu, India, and also Jepara. However, our content less than those reported from study conducted in Sempurna, Malaysia. From here, we can say that our phenolic content is within the range of reported studies for Sargassum polycystum. And let's move on to the infrared spectroscopy data. From here, please pay attention to the 5 main functional groups represented by 5 mark with number. We start from the with number around 3,300 represented for hydroxyl stretching. From here, we can say that our extract could has hydroxyl group, and then let's sum up to the aromatic ring with number around 1,600. This may indication that we may has aromatic ring represented by the stretching of double bond, carbon double bond here. And then we next look at the 4th WIP number corresponding for the COH bond. This may an indication that our hydroxyl the oxygen of our hydroxyl linked to the carbon in the aromatic ring. And also the last WIP number corresponding the CH aromatic bonding, that this WIP number confirm the aromatic ring and also the any substituted phenyl in this compound. And then the last one, we look at the second WIP number, confirming that there is a CH with sp3 hybridized. This may indication that this compound may has alkane as the substituents. And next, we need to compare with some report for fluorotannins, which are the phenolic compound from Sargassum. From here, we can see that this finding is in line with those reported for 5 prominent functional groups for fluorotannins. And this time is for the conclusion. To end this presentation, we could say that the Sargassum living in lung seizure can be thought as a potential antioxidant. And then by this study, we can confirm that this plant also can be considered as the phenolic source. And then by conducting infrared spectroscopy, we confirm the presence of hydroxyl, aromatic, and also alkoxy groups. And to close this presentation, I'd like to acknowledge the support from Pusat Penelitian dan Penerbitan UIN Mandiri and also Balai Penelitian dan Teknologi Bahan Alam LIPI Gunung Kidul for facilitating this study. And then here is the reference we use in this presentation. And thank you for your attention. You're welcome to give me any feedback as well as questions. Thank you.
Okay, thank you for your nice presentation, Mr. Anjar. And we are going to go to the discussion part. Now we have two questions. The first question from the Mrs. Risna to Mrs. Sofa. Has the alpha glucosidase inhibitor activity of this compound been previously recorded? And she gave the suggestion to study the activity relationship study of xanthan compound on alpha glucosidase inhibitory activity. So I will give the time for Mrs. Sofa to answer the question. Thank you.
Thank you, Ms. Faiza. Thank you, Gurisna, for the question. First, the question is, has the alpha glucosidase inhibitor activity of this compound have been previously reported? The alpha glucosidase inhibitor activity of this compound are not yet reported before. This compound founded in the 1999. He done about the atosinincy for testing against alpha salina, Artemisia salina as the cytotoxic activity. So also this compound is the first report on this from these plants. And thank you for the suggestion. Maybe the next research will be going to the study for the activity retention of the xanthan compound from the Artocarpus elasticus plants for alpha glucosidase inhibitor activity. Thank you.
Okay, thank you for your answer, Mrs. Sofa. How about from the Mrs. Risna? Is there any question or comment or something already clear or not? Okay, I think it's already clear, and we are going to the next question from Mr. Eka to Mrs. Tenny. The question is, if one compound has high activity based on molecular docking or meta-analysis, what the percentage in reality while this compound testing in vitro or in vivo? It's possible absolutely also high activity or not? Thank you. I will give the time for Mrs. Penny to answer the question, please.
Okay, thank you, Ms. Faiza. Thank you, Ms. Eka, for the interesting question. Here, I would like to try to answer the question. The prediction of active compound by molecular docking have accuracy about 60 up to 80%. So, it's possible the accuracy of molecular docking significant with the in vitro or in vivo assay. But if we can go to get the significant or closer with the in vitro or in vivo assay, we can continue the process of computational process simulation using bimolecular dynamic. Because in this method, in this molecular dynamic, we counting the environmental condition such as temperature, solvent, pH, and so on. But for example, I have done molecular docking and in vitro using active compound amide-cinnamide derivatives. have a significant correlation for anti-cancer MCF7. This is we have to get a correlation about IC50 of prediction and IC50 of experimental, and this sample for amide-cinnamide derivative for MCF7 genes have a good correlation for the molecular docking. So if if the possible, yes, it's possible. So accuracy for molecular docking about 60 up to 80%. Thank you.
Okay, thank you for your answer, Mrs. Tani Arnawati. I will ask Mr. Eka, is it already clear or you have any suggestion or comment for her answer?
Yes, thank you. I think it's clear. Yeah,
Okay, thank you.
Thank you.
And is there any question for our speakers? Maybe comment or suggestion for the speakers? Okay. I think we have no more question. And yeah, I will wait. Maybe some question will pop up in participant mind suddenly. Okay. I think no more question, and I will close this parallel session. You could join the next parallel session at 15:15. And I would like to thank for all of you for joining this parallel session. Hope you get useful information from this parallel session. Have a nice day and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Faiza.
Thank you too.
Good evening to all the participants. A bit of announcement to all of those who want or who wish to remain on this room. Please just stay on this room, or if you wish to change the room, please use your chat box or your raise hand icon to talk to the host. And we will have a break, and we will continue to the next session, the second session in 3:30 p.m. So 30 minutes from now. So we will have a break for 30 minutes. So please join us and come back again in 30 minutes. Thank you very much.
Ya.
Setengah 4 kan ya, kita mulai lagi ya?
5:15.
Apologize. We will have break for 15 minutes. So we will come back again to this room in 3:15. Okay, thank you very much. So 15 minutes from now. Thank you. Thank you to the moderator.
Is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berke America has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Berke America is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Berke America to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia. is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berke America has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Werken Jaga Medica is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Werken Jaga Medica to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia. Medica is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Werken Jaga Medica has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Werken Jaga Medica is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Berkepribadian America to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia. Is one of the most dynamic and innovative companies within the Berke Group. The Berke Group consists of over 35 operating companies in over many industries, including technology, infrastructure, and engineering. Berkepribadian America has been in business for over 17 years and has established itself as the major player in the research and the chemical analysis industries. Its customers include many bodies in the government sector, as well as large corporate entities. And its industries include food research, agriculture, petroleum, chemical, environment, and clinical research. We believe that Berkepribadian America is very well suited to meet with all stakeholders to support the quality of life and to improve it in Indonesia. We fully support Berkepribadian America to meet with all stakeholders and form partnerships to improve the quality of life of people in Indonesia.
Good afternoon, all the speakers and participants. Welcome for the parallel session 2, ISAP 2020, the day 1 in the room D with the topic of natural product and medicinal chemistry. Can you hear my voice clearly?
Yes, clearly.
Yes.
Yeah, thank you.
Okay, good. Before we start this session, I would like to check whether all the speakers already been here. So for Mrs. Halimatu Diana Nasution, Mrs. Halimatu Dahliana Nasution, already here? Not yet? Okay. Mrs. Praptiwi?
Yes,
here. Mrs. Sri Sugiwati also already here. Mr. Abdi Weraseptama?
Yes, I'm here.
Okay, good. Mrs. Megawati? Okay, and Mr. Arthur Aryola. Mr. Arthur? Already here? Not yet? Okay. Since our first speaker is not here, we are going to start with the second speakers. First of all, I will introduce myself. My name is Lucia, and today I will lead the discussion as a moderator, and I will try my best to keep this program on time. So in this afternoon, we will have 6 interesting talks, and every speakers will be given 10 minutes for presentation via the video recording, which we already received from the speakers. And after all the presentation finished, we will have a Q&A sessions. Also, I would like to remind you to not forget to fulfill the list of attendance that have been given by the organizing committee in the chat room box. And then the chat room is also available for delivering questions, requesting for changing room, and for the announcement given by organizing committee. So if you wish to change the room, please let the co-host know. We have Miss Dilani as a co-host. She will assign you to your chosen room if you wish to move to another rooms. So for the Q&A, you can deliver your questions in the chat room with you can write down your name and then your institution, and then to whom you would like to refer your questions and also your questions. Also, during the Q&A session, you can simply use the raise hand feature at the bottom of your screen if you wish to ask the question directly. So without further ado, we will start for the second speaker, a video from Mrs. Praptiwi. In this video, she will deliver a talk about evaluation of aphrodisiac activity of yellow ginseng, Renelia elliptica cord, root ethanol extract in DDY male mice. So for the committee, please kindly play the video.
I would like to present our study on evaluation of aphrodisiac activity of yellow ginseng or Renelia elliptica root ethanol extract in male DDY mice. The background of the study is sexual dysfunction could result in medical, social, and reproductive problems, and its prevalence is higher in men than women, but it could be managed by an aphrodisiac agent. Several synthetic compounds have been used as aphrodisiac agent, but it may have side effects such as retinal defects. However, several plant species are also known and used as aphrodisiac agent. One of these plant species is yellow ginseng. Renelia elliptica or yellow ginseng belongs to Rubiaceae, and it has local name in Minangkabau as jarum-jarum betina, kayu kuning, and kayu gemik. but in Malaysia it called segemuk and mengkudu rimba. The distribution of this plant is southern Myanmar, West Malaysia, Sumatra, and Borneo, and it has traditionally used to treat diarrhea, fever, cancer, postpartum, and aphrodisiac. So the aim of the study is to determine the aphrodisiac activity of yellow ginseng extract on sexual behavior of male DDY mice. Root of yellow ginseng was collected from Kalimantan and it was extracted with ethanol. The experimental animals for in vivo testing were DDY mice weighing 35 to 45 grams, 6 to 8 weeks old, that were obtained from National Veterinary Drug Assay Laboratory in Gunung Sindur, Bogor. The study has been approved by the Ethics Committee of the National Agency of Drugs and Food Control in Indonesia. The mice for this in vivo experiment were adapted for 1 week before treatment, and the mice were consisted of 5 groups, which are negative control administered with NaCMC 1%, and then positive control administered with sildenafil citrate, and then yellow ginseng extract at the dose of 50 milligram per kilogram body weight, 100 milligram per kilogram body weight, and then 250 milligram per kilogram body weight. And the duration of administration is 3 days, 5 days, and 10 days. Prior to pairing, the female mice were injected with estradiol benzoate 48 hours prior to pairing and progesterone 4 hours prior to pairing. After that, 1 male mice and 1 female mice were put in 1 cage, and then the sexual behavior of male mice were observed by video camera. The data were analyzed by analysis of variant followed by Duncan multiple range test. The parameter of sexual behavior included MON latency, MON frequency, intromission latency, intromission frequency, and ejaculation latency. The result of extract administration of yellow ginseng on MON latency showed that the administration of extract dose of 250 milligram per kilogram body weight shortened MON latency at 3 days administration, but at 5 days and 10 days, it is not significantly different from negative control. It might need higher extract concentration for administration more than 3 days. The shortened MON latency means improved sexual response. The effect of yellow ginseng extract administration on MON frequency in DDY male mice show that extract dose of 250 milligram per kilogram body weight increased MON frequency at 3 days administration, as well as on 10 days administration compared to negative control. Increased MON latency frequency means increased libido or sexual motivation. And the effect of yellow ginseng extract administration on intromission latency show that intromission latency at the administration of 250 milligram per kilogram body weight of yellow ginseng extract shorten the intromission latency, and the shortened intromission latency means fast sexual response. And the effect of yellow ginseng extract administration on intromission frequency, so there is no significantly different for all the treatment. Increased intromission frequency means increased sexual motivation. And the effect of ejaculation latency from yellow ginseng administration show that the administration of yellow ginseng extract prolong the ejaculatory latency at extract dose of 250 milligram per kilogram body weight at 10 days administration. Prolongation of ejaculation latency indicates an aphrodisiac action. The increase of ejaculation latency suggests as an indicator of improving sexual behavior. So the conclusion of this study that the administration of yellow ginseng extract at the dose of 250 milligram per kilogram body weight enhanced the sexual behavior as an indication of aphrodisiac activity. The administration of extract for 10 days consecutively prolongated the ejaculation, though it is not statistically different from the positive control. Thank you.
Okay, thank you very much for Mrs. Pratiwi for the presentations, for the video presentations. Now we are heading to our next speakers. We are going to hear a talk from Mrs. Sri Sugiwati. In the video, she will deliver the talk about antioxidant activity of liquid smoke fraction from kaju twigs and rice hulls. For the committee, please kindly play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Today is my pleasure to participate in this conference, the 7th International Symposium on Applied Chemistry 2021. I'm Sri Sugiwati from Research Center for Chemistry, National Research and Innovation Agency. I'm here to present our research with the title of Antioxidant Activity of Liquid Smoke Fraction from Kacepu twig, Melaleuca leucadendron, and rice heart Oryza sativa. This research was conducted with collaboration of Research Center for Chemistry, National Research and Innovation Agency, and Research Unit for Natural Product Technology, National Research and Innovation Agency. Liquid smoke is produced by condensing wood smoke during the pyrolysis process on wooden materials that have cellulose, hemicellulose, and lignin. Biomass containing high cellulose, hemicellulose, and lignin can be used as raw material to manufacture liquid smoke through the pyrolysis process. Kacepu twig and rice heart are biomass that contains high cellulose, hemicellulose, and lignin. This is the percentage of cellulose, hemicellulose, and lignin in Kacepu twig. And this is the percentage of cellulose, hemicellulose, and lignin in rice heart. This biomass can be used as substrate to pyrolysis process and produce crude liquid smoke. In the pyrolysis process, cellulose is decomposed into carbonyl and furan compounds. Hemicellulose is decomposed into acetic acid and carbon dioxide, while lignin is decomposed into phenolic compound. The main components of liquid smoke are phenol, such as phenol, guaiacol, and syringol, organic acid, such as acetic acid, propionic acid, and carbonyl, such as hydroxylbutanone, hydroxyl acetylaldehyde. These compounds are responsible for the flavor, color, antimicrobial, and antioxidant properties of liquid smoke. In addition to this compound, crude liquid smoke contains unfavorable compound polycyclic aromatic hydrocarbon PAH that can cause carcinogenic effect for human. In this research, we use vacuum evaporator method at different temperature to remove polycyclic aromatic hydrocarbon. The purpose of this research was to evaluate antioxidant activity against the PPH free radical scavenging of liquid smoke fractions derived from kacaput twig and rice heart. Evaluation of antioxidant activity of liquid smoke fraction need to be carried out so that it can be used as a natural food preservative, especially as a substitute for synthetic and conventional preservative that harm for health and environment. Materials. This study used liquid smoke fraction from kacaput twig, CT40, CT50, and CT60, and rice heart RH40, RH50, and RH60. Kacaput twigs biomass is obtained from byproduct of the kacaput oil refinery. Reagent 2,2-diphenyl, 1-picril hydrazine, quercetin, and methanol. Method. About 5 kg of kacaput twig and 5 kg of rice hearts were fed into the pyrolysis reactor. Pyrolysis process is conducted at 500 degrees Celsius for 8 hours. Liquid smoke resulted from the pyrolysis was collected in a container and left for 1 day to precipitate the tar. Liquid smoke resulted are brown color and contain PAH. This crude liquid smoke was then separated into 3 fractions using a rotary vacuum evaporator with the water bath temperature set at 40, 50, and 60 degrees Celsius for 1 hour at each temperature under vacuum condition at 200 millibar. The separation process resulted in liquid smoke fraction called CT40, CT50, CT60 for kacaput twig and RH40, RH50, and RH60 for rice hearts. After that, all of the fractions were stored in a freezer at -18 degrees Celsius to keep their stability. And then antioxidant activity of this fraction was determined by DPPH radical scavenging method. This is rotary vacuum evaporator that we use in our research for separation of crude liquid smoke. Liquid smoke fraction derived from separation process are clearer than crude liquid smoke with brown color. The liquid smoke fraction from separation process at water bath temperature of 60 degrees Celsius have higher yield than the yield of liquid smoke with the separation process at water bath 40 degrees Celsius and 50 degrees Celsius. The yield of RH60 is 41.4% and CT60 is 37.6%. After the liquid smoke fraction with a water bath temperature of 60 degrees Celsius is produced, there is no more evaporation and resulted black residue that contains PAH. This is the chemical composition of liquid smoke fraction derived from rice heart and kacaput twigs based on GCMS analysis. In each fraction derived from CT and RH contain acetic acid, phenol, guaiacol, and metacresol. In RH40, there are other phenolic compounds, namely cresol, phenol 2-ethyl, and phenol 4-ethyl 2-methoxy. This is the result of antioxidant activity of liquid smoke fraction by DPPH method. IC50 value of liquid smoke fraction derived from CT are 33.8 to 44.56 ppm, while IC50 value of liquid smoke fraction derived from RH are 26.7 to 41.4 ppm. Based on this IC50 value, liquid smoke fraction derived from CT and RH is classified as strong antioxidant activity. CT60 has higher antioxidant activity than CT40 and CT50 with an IC50 value of 33.8 ppm. Meanwhile, the antioxidant activity of liquid smoke fraction from right heart RH40 has higher antioxidant activity than RH50 and RH60 with an IC50 value of 26.07. The strong activities of liquid smoke fraction were caused by phenolic compounds which consist of phenol, phenol 2-methoxy, phenol 3-methyl, phenol 2-methoxy-4-methyl, phenol 2-ethyl, phenol 2-ethyl 2-methoxy. The antioxidant properties of the phenolic compound is caused by hydroxyl group on benzene ring. Phenolic compound in liquid smoke is produced by lignin decomposition at 500 degrees Celsius. This is a graph percent inhibition of DPPH radical scavenging by liquid smoke fraction derived from cacheput twig. CT60 have higher percent inhibition than CT40 and CT50. And then this is a graph percent inhibition of DPPH radical scavenging by liquid smoke fraction derived from right heart. RH40 have higher percent inhibition than RH50 and RH60. Conclusion. Based on the IC50 value, liquid smoke fraction from cacheput twig and rice heart was classified as a strong antioxidant against free radical DPPH with an IC50 value of 33.8 ppm for cacheput twig CT60 and 26.07 ppm for right heart RH40. Liquid smoke fraction derived from cacheput twig and rice heart are potentially developed as a natural food preservative, especially for smoking preservative of fish. Thank you very much for your attention.
Okay, thank you, Mrs. Sri Sugiwati, for the presentations. Before we continue to the next speakers, I wonder whether Mr. Arthur and Mrs. Halimatu Dahliana already joined us. Mrs. Halimatu Dahliana? Not yet.
Mrs. Halimatu Dahliana is in my team.
So, are you going to do the presentation? No, I mean, are you going to replace her in the session?
Yeah, I represent. I am the presenter.
Okay, good. Since we already started, so we will play your video for the last video. Is it okay?
It's okay.
Okay, good. So, and how about Mr. Arthur? So,
My name is Serlan, represent of Mr. Arthur.
okay, good. So all the speakers already here. Yeah? So okay, we are going to move. We are moving to the next presentation from Mr. Abdi Wiraseptama. In this video, Mr. Abdi will deliver a talk about the synergistic action of curcuma santoriza essential oil with tetracyclines against Pseudomonas aeruginosa and its effect on bacterial membrane permeability. For the committee, please kindly play the video.
Good afternoon, everyone. I'm Abdi Wiraseptama from Research Center for Chemistry Brain. On this occasion, I would like to present our research entitled Synergistic Action of Curcuma santoriza essential oil with tetracycline against Pseudomonas aeruginosa and its effect on bacterial membrane permeability. Antibacterial resistance has increased over the past few years, has been recognized as a major medical challenge in most healthcare settings. It has been known that World Health Organization recognized antimicrobial resistance as one of three greatest threats of human health. And the major reason of resistant antibiotic is inappropriate use of antimicrobial drugs. It brings impact of antibiotic resistance such as increase of treatment failure, increased cost of treatment, increase of mortality, and poor patient outcomes. There are four major mechanisms of antibiotic resistance that can reduce the effect of antibiotic toward any pathogen. For example, alteration of drug target, inactivation of drug by the enzyme that can reduce the activity of antibiotics, activation of drug efflux pump, as well as inhibition of drug uptake by decreasing the permeability of membrane cells. There are common strategies that can be used to overcome bacterial resistance. For example, discovering new antibacterial from alternative source, for example, natural product, and the use of combination of drugs, for example, the use of antibiotic altogether with the antibacterial agents from natural products. Natural products always become a potential source for discovery of antibiotic. Curcuma santoriza, for example. This plant has been used in folk medicine in several countries for the treatment of infection disease. This plant also contains essential oil, which has been reported to possess several biological properties, for example, anti-inflammation, antibacterial, immunomodulator, anti-cancer, etc. Although the antibacterial activity of essential oil has been reported earlier, however, there is no report about the synergistic of combination of essential oil antibiotic against any pathogen. Therefore, the objective of this study are to investigate the interaction of curcuma santoriza essential oil in combination with tetracycline against Pseudomonas aeruginosa, as well as to evaluate its mechanism of action on membrane cell permeability. Well, for the first assay, we used checkerboard method. This assay was determined to know the any synergistic effect in combination of essential oil and antibiotic. The fractional inhibitory concentration index will be interpreted as synergistic effect if FICI less than 0.5 and antagonism if FICI more than 4. From this result, we can see that the combination of essential oil and tetracycline produce a synergistic effect with FIC index less than 0.5, that is 0.25. In this combination, we can see the MIC value of tetracycline in combination with essential oil was reduced 1.9 something. And from this result, we can see that essential oil and enable to reduce the MIC value of tetracycline in combination. In contrast, in combination of essential oil and ampicillin only produce indifference effect with FIC index 1 point something. In this combination, MIC value of ampicillin in combination and MIC value ampicillin when used alone was the same. In order to know the mechanism of action of this combination on membrane cell permeability, the bacteriolysis activity was then conducted. In this study, crystal violet uptake or percentage crystal violet uptake was determined to know any activity on membrane cell permeability. And the percent of crystal violet was determined using a micropill reader at 590 nanometer. From this figure, we can see that the combination of essential oil and tetracycline was higher in the uptake of crystal violet when compared to essential oil used alone as well as tetracycline when used alone. So in this study, we can see that tetracycline and essential oil give the high activity to produce bacteriolysis activity, as well as the essential oil also enable to give the potency to tetracycline to kill the bacterial pathogen. Next assay is loss of 260 nanometer absorbing material. This assay was conducted to know the effect of the release any material, intracellular material inside the cell and lead the cell lysis. In this study, any absorbing material at 260 nanometer was observed using micropill reader at 260 nanometer. From this result, we can see that the combination of essential oil and tetracycline produce high activity at OD 260 nanometer. That means that this combination can produce the activity to lead bacteriolysis compared to essential oil when used alone, as well as tetracycline and used alone. So in this combination, we can see that essential oil may enhance the activity of tetracycline to kill Pseudomonas aeruginosa by lead to lysis activity. So the conclusion of this study is essential oil from Curcuma xanthorrhoea enhance antibacterial activity of tetracycline with a synergistic effects against Pseudomonas aeruginosa. The combination of essential oil and tetracycline enable to kill Pseudomonas aeruginosa through the disruption membrane cell permeability and induced cell death. And the essential oil of Curcuma xanthorrhoea may be used as adjuvant of tetracycline to overcome the pathogenesis of Pseudomonas aeruginosa. We would like to thank the National Priority Research Grant, Research Center for Chemistry, and Center for Development, Education and Training. Thank you for your kind attention.
Okay, thank you, Mr. Abdi, for the video presentations. Next, we are going to hear the presentation from Mrs. Megawati with a talk entitled Phytochemical Screening and Free Radical Scavenging Activity of Cordersiodendron pinnatum bark extract. For the committee, please kindly play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Thank you for allowing me to present our result in this conference, with the title is Phytochemical Screening and Free Radical Scavenging Activity of Cordersiodendron pinnatum bark extract. My name is Megawati. I'm from Research Center of Chemistry, Brain. Research of the chemical content and free radical scavenging activity from Cordersiodendron pinnatum bark has been conducted. The bark of Capinatum was examined for phytochemical screening and free radical scavenging activity by diphenyl 1-picrihydrazyl assay. The result on phytochemical screening show the both extract and fraction contain flavonoids, saponin, and tannin. The ethyl acetate extract has phenolic content 30.77% and highest free radical scavenging activity with IC50 5.20 microgram per milliliter. Vacuum liquid chromatography was carried out from the ethyl acetate fraction and obtains X fraction. Subfraction 6 has the highest free radical scavenging activity with IC50 9.21 microgram per milliliter, with the positive control persisting for 4.43 microgram per milliliter. These results suggest the Capinatum merbach extract have strong antioxidant potential to inhibit free radical. The introduction is the free radical scavengers such as peroxide, hydroperoxide, or lipid peroxide can inhibit oxidative mechanism cause degenerative disease. Process of the oxidation reaction free radicals circumstance as environmental stress, disease, pathogen attack increase beyond normal level. Increased reactivity cause the free radical compound attack healthy cell in the body cause the cell lose their function and structure. Natural antioxidant is a noticeable interest in antioxidant because several alternatives as medicine. Pinatum merbach is a lac deciduous tree. This plant is widely distributed Philippines, Kalimantan, Sulawesi, Maluku, and Papua New Guinea. Pinatum merbach extract contain bioactivity compounds such as saponin, flavonoid, and polyphenol. This plant also shows diverse range biological activities, including broad-spectrum antibacterial activities and free radical scavenger activities. The ethyl acetate of Pinatum merbach has antioxidant activity with IC50 89.40 ppm, and the methanol extract with IC50 234.5 ppm. The further research on the bioactivity of radical scavenging from methanol extract of Pinatum merbach has not been carried out. Our preliminary results show the ethanol extract has been performed its antioxidant activity with IC50 170.86 ppm. The purpose of this study were to examine the phytochemical and free radical activity of the methanol extract of Pinatum merbach. The methodology of research is the dry of Pinatum merbach was cut and milled into small pieces, 100 grams, and extract by 2.5 liter methanol at room temperature and for 3 days. And then the methanol extract is evaporated under vacuum to obtain crude methanol extract. The crude methanol extract was fractionated with the hexane, ethyl acetate, and butanol. And the ethyl acetate extract was further separated using vacuum liquid chromatography with gradient elucidation, and hexane, ethyl acetate, and methanol. We get the egg subfraction, and all extract subfraction and subfraction were evaluated the free radical scavenging activity, phytochemical, and total phenolic content. With the free radical scavenging activity with the PPH method, 4 milligram was this 4 milligram for sample dissolved in the methanol 4 milliliter. The concentration variation we prepared at with the 0.5 milliliter of 1 millimolar DPPH in the methanol. The mixture was incubated in the dark for 30 minutes to the absorbent 517 nanometer. This is extent of the color. For the total phenolic amount of 5, 10, 15, and 200 microliter from 1000 ppm of gallic acid solution were pipetted and placed into test tube. Each tube were added with the 1 milliliter of the sample and distilled water to 7.5 milliliter volume. Add with 0.5 milliliter to foreign culture reagent and mix thoroughly for 8 minutes, followed by the additional 1.5 milliliter of 20% sodium carbonate. The mixture was allowed to stand for further 60 minutes in the dark, and absorbance was measured at 765 nanometer. This is extent of the color total phenolic content. Result of the result after all The macerated was collected the solvent separated by concentration in using a vacuum rotary evaporator. The methanol extract was dried at 40 degrees Celsius and obtained dry methanol 146.21 gram, and the yield is 18.27%. The phytochemical screening was carried out for its extract and function and preliminary analysis to determine the class of compound contained in the plant, so its utilization could be estimated. That the extract and fraction of the stem bark Capinatum mare contain to flavonoid and saponin. Flavonoid content in the bark of Capinatum mare is one of the factors that influence the antioxidant effect. TPC active process throughout the amount of the component in the plant have rare properties, and the properties allow the acting as antioxidant. The ethyl acetate extract exhibit higher TPC as compared to the Al fraction with the range 28 until 30. Hexane extract with approximately 4 until 5, and butanol extract 25 until 27. Higher polyphenol content in the ethyl acetate extract is responsible for bioactivity. Therefore, this extract is expanded to exhibit good result in the free radical scavenging activity. For antioxidant result for antioxidant activity, scavenging activity of DPPH is based on 1 electron reduction with the rest present the free radical reducing activity of antioxidant. Quercetin 4.43 were used as positive control. All the sample were run in triplicates. The results show the presented inhibition for free radical of the Capinatum extract standard at different concentration. The lowest IC50 was detected is the ethyl acetate fraction 5.20 extract followed by methanol extract 9.80 and butanol extract 7.84. As the lower IC50 value possesses a higher antioxidant activity. The ethyl acetate extract has higher ability to scavenge free radical compared to methanol, butanol, and hexane extract. Highest antioxidant activity shown by the ethyl acetate extract has positive relationship with TPC activity, where high TPC give a high antioxidant capacity due to the linear correlation between the two parameters. Subfraction 1 and until fraction 4 with the subfraction ethyl acetate fraction have not the potential as free radical scavenging activity because the IC50 value is very high, more than up to 100,000 microgram per milliliter. And subfraction 5 until subfraction 8 is very strong because the value IC50 is less than 50 microgram per milliliter. And the test result are each subfraction show that subfraction 6 has the greatest and the free radical scavenging activity of the other subfraction with the small IC50 9.21 ppm. The conclusion, the phytochemical screening revealed the presence of phenolic group may relate the free radical scavenging activity of Capinatum mare. The use of Capinatum mare as natural antioxidant appear to be alternative herbal medicine. That's my presentation. Thank you for your attention.
Okay, thank you, Mrs. Megawati, for the presentation. Before we go to the next presenter, I want to encourage all the participants to drop your questions in the chat box, so later we can have a good discussion after all. And for the next presenter, we have Mr. Arthur Ariola Lono. In the video, he will deliver a talk about screening of astaxanthin active compound from lithopenous vanami shrimp shell using HFC 134A subcritical extraction. For the committee, please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. My name is Berdaulat. Now I will present our research about the screening of astaxanthin active compound from the stem cell using FC 134A synthetic extraction. Mr. Arthur Aryo Lelono as a major author of this research, and the other author are Erlangga Dewan, Sabar Semanung Khalid, Dedi Sukanda, from Research Center for Chemistry, and Faculty of Science and Technology in San Diego. Okay, shrimp cell is a stay of the ocean fisheries. About more than 900 tons emission has been produced shrimps according to the data of Central Bureau of Statistics in 2017. It product is for frozen, headless or headless or tail shrimps, and it will cost the Western shell about 60 and 70% of the shrimp to produce Western shell. If we can handle it inappropriate of this Western shell shrimp, it will a negative impact for the environment, such as bad odor, synthetic compound, increasing density, and threaten to the ecosystem. The utilization of this Western shell so far is only for feeding animals and crackers. It only has low economic value. So, how to overcome this Western shell to increase the economic value? So we can producing the carotenoid pigment about astaxanthin from Western shell. What is the astaxanthin? The astaxanthin is the carotenoid pigment in sample group found in fresh microorganism and marine animals, and has been more benefit than the others of compound. It's 600 stronger than system MC. 550 times stronger than A, boost your energy and use apnea and prevent the organism, increasing their focus and reduce muscle soreness and hence excessive focus. In this research, we carry out the extraction using of FC134A synthetic extraction system with the tetrafluoroethane as a material solvent for this apparatus from this system extractor. The material will extracted by the gram about 50 about the 250 gram Simplicia option shell. We'll put into the extraction chamber and the extracts will collect in the collector chamber. The process it will take the 1 hour and 3 hours. The result of this research can you can see in the table 1, the extraction process of L-Panama stem cell with the FC134A synthetic system. We have the various of the sample total ratio 1:5, 1:10, and 1:20. The highest yield is in ratio sample solvent in 1:30, 1:20 with time of the process about 3 hours. Its yield is 1 gram of extract, and the extract percent is 1.5%. The selective of the highest yield of the ratio and 1:20 will it will be verified. It will verified with column chromatography, and it's resulted 20 fraction. You can see in the table 2. And in figure 1, this is the extract of the stem cell. And figure 2, this is the TLC of Panama stem cell with variation of the mass ratio sample. You can see in the figure 1 and the figure 2 the spot with the white color. Spot with white color is parallel with the standard of the assessment. In figure 3, this is the antioxidant qualitative assay of the fraction F, fraction 2, fraction 4, and fraction 5, which is the which has positive result marked by the presence of the white spot in the figure 30. In the figure 3A, we can see that it has a white spot. It's shown that this is in this sample is have the antioxidant activity. For the FTA spectrum, from the fraction 2, fraction 4, and fraction 5, you can see in the figure 4, there is a lot of functional group. You can see functional group in the table 3. This is the list of the functional group analysis. Again, the external group is the OH, CH, and carbonyl, hydroxyl, methyl, alkanes, and ethylene, and methyl. Based on the FAM spectrum data, the fraction 2, fraction 4, and fraction 5 indicate the presence of several characteristics of the therapeutic compound functional group. The different attribution strength of several functional groups indicate the impurities of this iterative fraction. And then this is the result of the LCMS-MS analysis. You can see in the figure 5A. This is the chromatogram of the astaxanthin standard. You can see the retention time for astaxanthin standard in the 10.16 minutes in the 10.16 minute, and the mass spectrum of the astaxanthin at low energy molecular ion is in the 597 with a fragment in one with a fragment of product ion at 147 grams. This is the characteristic of the astaxanthin. In figure 5C, we can see that, in the figure 5B, we can see that this is the chromatogram of the fraction 5. It has several peaks. The peak of the retention time of the fraction 5 is at the 30 in the 10.16 minutes, and the mass spectrum of the fraction number 5 is in 597.4, and the product ion is the 1.7. From this, according to this result, the fragmentation ion product show that is coincident with the retention time of the standard astaxanthin. In the figure 5B, we can see that has result several chromatogram appear at the retention time 798, 923, and 1044, and 11:21 minutes. The peak show that is a relative small amount in the target compound, the result of astaxanthin. It's relative to the feed of shrimp culture and found compared to natural shrimp. Through the database of the analyzed LCMS, the fraction 5, their molecule weight reveals several potential compounds identified. It's shown in table 4 in the retention of the 7 and 8. The compound is oxyclean, dolichosterone, cholecalciferol, corticosterone. And the figure 6, this is the structure of the astaxanthin. So the conclusion is the astaxanthin compound from the alkaline shrimp has West stream was extracted using separated extraction of FC on 34A based on the result of SMS extraction. 5, which show the same retention time as the extension standard. At the retention time 10, 16, other compound from each fraction tested, fraction 2, fraction 4, fraction 5 had an antioxidant activity based on the qualitative test of antioxidant activity. Other compounds are suspected to occur in a selected fraction, namely antioxidant, dolichosterone, and D3 corticosterone. Thank you for the attention. Assalamualaikum warahmatullahi wabarakatuh.
Well, thank you. Thank you, Mr. Herlan, for the presentation. And I apologize that I mentioned other people's names instead of your name as a presenter. Well, last but not least, we are going to hear the presentation from Miss Gita Wulandari. In her presentation, she will talk about the tensile properties and antimicrobial activity of bioplastic based on sagu starch utilized fiber betel leaf. For the committee, please play the video.
Hello everyone. Now I'm going to present our research entitled Tensile Properties and Antimicrobial Activity of Bioplastic based on sagu starch utilized fiber betel leaf. First, the negative environmental impact encourages the using of bioplastic. There are several sources of bioplastic, but starch is the most studied because starch is widely available and have good tensile properties. But starch-based bioplastic also have some disadvantages since it breaks easily and are resistant to oxidation and microbiological activities. So we have to add some additive to bioplastic like plasticizer and antimicrobial agent. Plasticizer addition can increase flexibility of the bioplastic film. Antimicrobial agent makes antimicrobial activity and make the bioplastic shelf-time longer. In this study, we use sagu starch as bioplastic matrix, glycerol as plasticizer, and betel leaf extract as antimicrobial agent. Betel leaf contains tannin and flavonoid that can inhibit the microbial growth. The aim of this research is to investigate the effect of betel leaf extract and glycerol addition on the mechanical properties like tensile and elongation at break, and antimicrobial activity of sagu starch bioplastic. The antimicrobial analysis was done by investigating bioplastic films inhibition on bacteria, Bacillus cereus, and fungi Aspergillus niger with this diffusion method. And here are some materials and equipment that used in this study. This research is start from the preparation of betel leaf extract. First, we make the betel leaf powder, and this betel leaf powder will be extracted by ultrasonic method, filtrated and evaporated by using rotary evaporator. The plastic film making was done by waving 10 grams of sagu starch and adding 100 milliliters of aqueous and various concentration of glycerol with 0, 10, 20, and 30% volume, and after that we add betel leaf extract with various concentration 0, 3, 6, and 9%, and after gelatinization process, this mixture casts on acrylic and wait until dry. Then the bioplastic film will be tensile, elongation at break, and antimicrobial activities. Figure 1 shows the effect of glycerol addition on stiffness and elasticity of bioplastic. The shortest extension was shown by bioplastic sample without any glycerol. That means it broke easily in a shorter time than bioplastic with glycerol. This shows that starch-based bioplastic properties, which was hard but brittle. Glycerol addition increased the elasticity of bioplastic. Figure 2 shows tensile strength of bioplastic. The increasing of betel leaf extract and glycerol composition will decrease the tensile strength. It's due to betel leaf extract and glycerol addition reduce the intermolecular forces of bioplastics. This disturbance will reduce the value of the tensile strength. Figure 3 shows the elongation at break of bioplastic. The more glycerol composition and betel leaf extract added, the more glycerol molecules bound to starch and tannins or flavonoid. This will cause the bioplastic to become more elastic and flexible. Glycerol molecules will increase the mobility of bioplastic and extensibility of the films. Figure 4 shows the antimicrobial activities analysis of betel leaf extract. The antibacterial activity show the diameter of inhibitory zone of betel leaf concentrated extract is bigger than betel leaf with 2% dilution. It means the higher tannin and flavonoid content makes the higher antibacterial activity. The antifungal activity of betel leaf concentrated extract shows the large inhibition. However, betel leaf extract with 2% dilution doesn't show any inhibition. Figure 5 shows the diameter of inhibitory zone of bioplastic. All variation of bioplastic show an antimicrobial activity. The higher betel leaf extract composition, the higher inhibitory zone diameter will be. More composition of glycerol makes the inhibitory zone larger. And bioplastic with 20% glycerol show the maximum inhibitory zone. Figure 4 shows the antifungal activities analysis of bioplastic with different glycerol concentration, and it showed that all variation of bioplastic didn't show any inhibition in antifungal activity. So, further studies are needed to find out the minimum concentration of betel leaf extract to against the Aspergillus niger. And the conclusion of this research are the highest tensile strength value was shown by bioplastic sample without any glycerol and betel leaf extract. The highest elongation value was shown by bioplastic sample with 30% glycerol variation and 9% betel leaf extract. All sample of bioplastics can inhibit the bacteria Bacillus cereus growth. The antifungal analysis show that only betel leaf extract concentrated can inhibit Aspergillus niger growth. And it is important to find the minimum concentration of betel leaf extract to inhibit Aspergillus niger growth by doing further researches. I think that's all. Thank you for your attention.
Okay, thank you for Miss Gita. Wait a minute. Thank you for Miss Gita for the presentation. And now we are moving to the Q&A session. I would like to ask all the speakers to open your camera so we can see your present. Okay, good. And I want to remind all the participants to not forget to fulfill the list of attendance that have been given by the committee in the chat room box. So let me see, we have several questions. For the first question from Mrs. Yeni Meliana to Mrs. Praptivi. Is the yellow ginseng root that you have used in your study is the same with the ginseng from Korea that we have commonly known? Is it the same ginseng from Korea? For Mrs. Praptivi, please answer the question.
Okay. Thank you, Miss Lucia, and thank you, Bu Jenny. Yellow ginseng in this study is different from Korean yellow ginseng. Korean yellow ginseng belongs to Araliaceae family, and it has scientific name Panax ginseng or Aralia ginseng, and it has a compound called ginsenosides. Maybe that's what.
Okay, no problem. We have another question from Mrs. Yeni to Mrs. Megawati. If we want to develop the active compound from sample to bigger scale, or should we extract it from the plant, or is there any other way, such as chemical synthesis?
Okay. Thank you for Dr. Melly for the question. This research is the only limited to the activity for the extract, and we'll continue to look for the active compound. If it does produce an active compound, it is possible to produce it using synthesis. But if the extract is still the producer for the extract, if it is difficult, the plant can be done with the tissue culture. Thank you.
Thank you, Mrs. Megawati. Is there a comment from Mrs. Yeni?
No, thank you, Mega, for the clearly answer. Thank you.
Okay, good. Thank you. For the next question, we have question from Mr. Eka to Mr. Abdi. Why do you choose Pseudomonas as the targeted bacteria? Is it only for the model gram-negative, or also could have similar activity in gram-positive bacteria? And could you suggest the compound interaction between essential oil and tetracycline during disrupting the bacterial membrane cells? Please, Mr. Abdi.
Okay, thank you, Ms. Dr. Eka, for the question. First, Pseudomonas aeruginosa. Why I use Pseudomonas aeruginosa? Because I interested with this bacteria. As we know that Pseudomonas aeruginosa is one of gram-negative bacteria that's difficult to control. And since the Pseudomonas aeruginosa have the unique mechanism of resistance against antibiotics. And also WHO is already listed 6 bacteria that have potential to be developed as superbug bacteria, and Pseudomonas aeruginosa is included as the 6 dangerous bacteria that can be developed to be superbug bacteria. That's why I use Pseudomonas aeruginosa as the model in this essay. But regarding the gram-positive bacteria, of course, we can use in this essay to know the synergistic, to know the interaction between our compound, our essential oil with antibiotics, something like that. Regarding the synergistic action, maybe it's interaction between essential oil and tetracycline probably work at the different site of action. Since we know the compounds, just like flavonoid and essential oil, most likely work on the membrane permeability that can decrease the permeability of membrane cell, that makes the tetracycline penetrate easily inside the cell, then make the tetracycline do the work at the site of action, like protein synthesis, something like that. So, essential oil and tetracycline have the synergistic action by different target of action, something like that. It's not additivity. Additivity is mean like one compound have the same target with another compound, something like that. So in this essay, in these results, we can conclude the synergistic action of between essential oil and tetracycline due to the different site of action. That's my my answer.
Thank you, Mr. Abdi. I think it's really clear. Are there any comments from Mr. Eka?
Oh yeah, I think it's enough. Yeah, it's clear.
Enough? Okay, good.
Thank you.
Let me see. Are there any other questions? No questions. So I have some questions for Ms. Gita. Ms. Gita?
Yes.
Oh, okay. The purpose of your study is to develop the bioplastic material, something like that, right?
Yes.
Yeah. I get the idea that the addition of the fiber beetle leaf extract is supposed to inhibit the growth of microorganisms in bioplastic. However, the presence of antimicrobial compound, I think it could seriously affect the biodegradation pattern of the bioplastic, since it can interfere the action of natural microbial population that's responsible for the biodegradable process. So what do you think about this issue? That's my first question. And my second question is, do you think bioplastic is a solution for the plastic problem we are facing right now?
Yes.
Please give me your answer. Thank you.
I think it's like a dilemma. One of in one side, the addition of antimicrobial makes the plastic shelf time become longer, but it also makes the bioplastic a little bit hard to degrade by microorganisms, but maybe at the right composition, it will be make a balance between two problems. And for the second question, bioplastic, maybe I think bioplastic is the solution to replace the commercial plastic from the synthetic plastic, because there are several advantages for the using of bioplastic. Thank you.
Thank you for the answer. But I think we also know that not all the bio-based plastic is biodegradable, right?
Yes.
Some of the biodegradable plastic is not based on the biomaterials. But I think it's a really good answer, and I agree with you. It's quite dilemmatic, right? So thank you for your answer. And we have another one question to you, to Ms. Gita, from Mr. Eka. It mentioned on the results that higher fiber beetle leaf extracts supplementation to bioplastic relevant with higher antibacterial activity. Could you suggest the maximal concentration should be applied, and why in the antifungal activity, the bioplastic combining fiber beetle leaf extract didn't have any activity? Please give your answer.
Wait a moment, please.
Sure, take your time. Oh, and is it okay if you want to give your answer in bahasa Indonesian, since all the participants are Indonesian anyway?
Yeah. Okay. I think I cannot suggest the higher the maximal concentration, because it needs further studies about it by trial. And yes. And for the second question, why the antifungal activity didn't show any inhibition in bioplastic? I think it's because I just know when sorry I changed bahasa. Jadi waktu saya penelitian ini nggak dicek dulu gitu ya. nggak melakukan penelitian terlebih dahulu mengenai berapa konsentrasi yang sesuai untuk antifungal activity. Saya hanya membaca jurnal gitu terkait dengan itu, dan itu pun bukan untuk spesies Aspergillus niger gitu. Dia untuk jamur, tapi saya lupa spesies jamurnya. Itu untuk jamur yang lain, dan saya langsung merujuk dari literatur tersebut untuk mengambil konsentrasi hambat minimum dan konsentrasi bunuh minimum dari 2% pelarutan itu, pelarutan 2%. Yeah, I think that's all. Thank you for Mr. Eka.
Okay, thank you.
For the question.
Are there any comments from Mr. Eka?
Yeah, I think it's enough.
Okay.
Thank you.
Okay. So, are there any other questions from the floor? No questions? So I'm going to ask Mr. Harlan. As we know, the shrimp shell is rich of protein and chitin inside this shrimp shell. And since the presence of protein and chitin can give the effect on the extraction process of what's the compound, the astaxanthin. I wonder whether you remove all the proteins and the chitin prior the extraction, and if you did that, what kind of method did you use to make sure that there are no damage with your targeted compound, in this case astaxanthin? Thank you.
Can we answer with bahasa?
Sure, go ahead.
Jadi untuk ekstrak ini kita dapat ekstraknya itu dalam bentuk pasta, dan saya belum pernah mencoba untuk memisahkan protein yang ada dalam ekstrak itu.
So you didn't do the extraction by yourself, then?
Yeah, yes.
Okay.
I don't know what way to separate its protein. I never tried.
But have you ever maybe read in some publication
no,
or maybe?
No, maybe later I will deliver to Mr. Arthur to answer this question.
Okay, thank you for your answer. Are there any questions from the audience? Questions? So just a quick and simple question for Mrs. Prativi. I wonder whether any special reasons on why you chose to employ DDY mice as a mice model. I mean,
excuse me, would you repeat the question, please?
Okay. I just curious on why you chose to employ the DDY mice for the mice model. I mean, are there any like a special criteria in select the species for your in vivo experiment?
No, because the in vivo study, actually we should use 3 species of experimental animal, and the DDY mice, it needs a smaller extract to use for the in vivo study. If we use rat, we use more extract, and we up to now we did not have a lot of extract of yellow ginseng.
That's all.
Yeah.
Okay, thank you. But I wonder whether we can replace the mice model. Maybe, a lot of mice strain, you can say maybe Winstar or Boxy or whatsoever strain. If the reason is because of mice need a small amount of extract, can we just replace the mice with another strain, another mice strain?
We can replace the species of the DDE mice with other strain. Yeah.
Okay. Okay, thank you for your answer.
Okay.
It's really clear. I'm still waiting for another question from Sean. Okay, one last question from Mrs. Sugiwat. I want to ask, in your presentation, you mentioned about the polycyclic aromatic hydrocarbon as a result of pyrolysis process. Yeah. You mentioned that you remove those toxic materials by vacuum evaporator, but I wonder whether there are any additional tests to make sure that all of those hazardous materials have completely gone from your samples. Thank you.
Can you pardon your question?
Oh yes, you mentioned about the polycyclic aromatic hydrocarbons, and you said this compound is carcinogenic. So, and then you also mentioned that you remove this compound by vacuum evaporator. I wonder whether there are any additional tests to make sure that these toxic materials, can say that, have completely gone from your fraction. Okay.
Thank you, Ms. Lucia, for your question. Polycyclic aromatic hydrocarbon is carcinogenic effect for human. In our research, we remove polycyclic hydrocarbon by separation process using vacuum rotary evaporator with the different temperature water bath at 40, 50, and 60 degrees Celsius. Crude liquid smoke that contain polycyclic aromatic hydrocarbon is brown color. And then after we separation process using vacuum rotary evaporator, the liquid smoke is clearer. In our research, we didn't check the polycyclic hydrocarbon content, but in many reference we can read that there are no polycyclic reduce the polycyclic aromatic hydrocarbon. There are many methods to separation process to remove PAH, distillation fractionation, and in this research we use rotary vacuum evaporator because we didn't use distillation vacuum because in the in my laboratory I have not this apparatus. Thank you.
Okay, thank you for your answer. So by observing the color of your samples, we can already know whether there are any what was the name? PAH inside your sample, right? by observing the color only?
Yes, I observed the color of the liquid smoke. Include they are brown color, and then there are many PAH, and then in liquid smoke result from separated process, they are clear color.
Okay, thank you for your answer. That's really clear. No questions from the floor? Is anybody want to ask something to our speakers? Or else I'm going to close this session. No question. Okay. Well, thank you very much for all the speakers and the participants for your participation in this oral session. It was fun, and I hope all of us gain benefit from this fruitful discussion. Before we close this session, I would like to remind you to not forget to fill the list of attendance that have been given by the committee in the chat box. And also, I would like to remind you that tomorrow we still have many interesting talks. The second day will be started at 8 a.m. And we have two keynote speakers in the morning. We have Dr. Anna Velan from University of Queensland, Australia, and Dr. Firman Kurniawansyah from 10 November Institute of Technology, Indonesia. So stay tuned and don't forget to participate via the Zoom portal. And also you can share this information to your colleagues since this event also will be aired via YouTube in the channel Lembaga Ilmu Pengetahuan Indonesia LIPI. So this is the end of our session. Thank you very much once again and have a good day. See you tomorrow. Goodbye. You can leave the room. Take care. Bye.
Thank you all. Bye-bye. See you tomorrow.
See you.
Thank you all.